Well, good morning, everybody. Good morning. Oh, it's nice to have a response. I'm Sarah Schlesinger. I'm a research associate professor here in the Laboratory of Cellular Physiology and Immunology and at the Aaron Diamond AIDS Research Center. And I'm delighted to welcome you to the 47th annual holiday lecture for high school students at the Rockefeller University. And this lectureship was established in 1959, which parenthetically was the year before I was born and long before any of you were born, by Dr. Alfred Mirsky. And he based it on a series of lectures that had been given in London in 1826 by Michael Faraday, who was largely regarded by the scientific community as one of the greatest experimentalists and greatest science teachers of all time. And it's remarkable to think in 1826 people were getting up to teach science to other people. Sort of amazes me. Dr. Mirsky believed that you got people into science by hooking them when they were young, and that's his terminology, not mine. And me standing here before you is a prime example of that, because 30 years ago, I first came to the Rockefeller to the holiday lecture in 1976 and sat in the audience as a high school student. So I feel particularly honored and a little shocked to be standing here on the podium to welcome you. And the lecture that I attended was given by Dr. Christian de Duve, who had won the Nobel Prize in 1974 for figuring out how the cell biology, the anatomy or the microanatomy of the cell. And the lecture I attended was called The Lives of the Cell. And subsequent to that lecture, he wrote a two-volume book series called The Lives of the Cell based on that lecture. Following the lecture, he thought about what he had talked about in a different way. And so I guess that's one of the things that I just wanted to mention to you, that you coming here, is to give you a glimpse into sort of real science, as it were, to what we do, but it also helps us to see, to hear your questions, to see your enthusiasm, your vigor, your youth. And in Dr. de Duve's preface to his book, Vital Dust, which I would recommend to all of you, he comments on how giving the lecture to us and the questions that we asked helped him to rethink his view of the cell, which sort of amazes me because here this guy who'd won the Nobel Prize for figuring it out and teaching us actually was able to learn something from the students. So with that, I would like to thank you all for getting up early on vacation, which I know is a big deal. Giving up a day of your vacation to hear about science is not a small feat. And I especially know this because I'm the mother of 4 boys. And one of my sons, who's a Hunter student, is here, and getting up is a big deal because my son, who's at Stuyvesant, didn't want to get up. So with that said, we're going to start our lecture, and I would just like to remind you of a couple of housekeeping things before we do. The first is there's no smoking or eating or drinking in the auditorium. If you have cell phones or beepers or anything that beeps, please make sure that they're off now. Everybody check. Thank you. And then when you come back, please make sure again. There will be lunch at noon, and I'll let you know about that at the end. And so with all of that said, I would like to introduce you to Dr. Leslie Vosshall, who is the head of the Laboratory of Neurogenetics and Behavior here at the Rockefeller, who's going to give our lecture, Covert Ops, the Chemicals Behind Invisible Communication. Leslie is head of lab here. She went to high school in New Jersey and then did her undergraduate work here in New York at Columbia and was a graduate student here at the Rockefeller. She then went back across town to Columbia to do her postgraduate work with Richard Axel, who won the Nobel Prize in 2004? 2004, for figuring out basically how we smell, which is just not smell bad, but smell with our noses. And so Leslie has come back here to head her own laboratory that works on not only how we smell, but how insects smell and how important that is in a whole bunch of things. And she's going to tell us all about that this morning. So Leslie, thank you.
Thanks a lot for coming out. I'm so excited. I'm so excited to give this talk. We have many, many things that we've set up for you. We're going to have audience participation, taste testing, smell testing, and just hours and hours of fun. Because I decided that you guys are probably an inherently competitive bunch, I thought that we would, in order to encourage, I don't want to stand up here for 3 hours and just talk and have you listen. We will divide the auditorium into the South team and the North team. We have Jennifer over here keeping score. It's slightly unfair because there's a few more people here on the edges, but So the way you get points is by asking a question. By raising your hand, someone will hand you a microphone. We'll have specific points where you guys can ask questions. I'll try to provide answers. Or if you have a huge burning question, please feel free to raise your hand and we'll get to you. So what about me? As Dr. Schlesinger said, I went to public high school in New Jersey and then spent the rest of my professional life in New York City, bouncing from the east side to the west side. Bottom line, to be a scientist, you have to spend a lot of time. So I spent 17 years. I worked in laboratories in high school, 17 years until I got my first real job, which I think is a pretty typical story for scientists. It is a life's work. You get really into it, and you spend years and years and years training. Now I want to find out about you guys. I assume that you're getting up during your vacation. You probably are interested in science, but I just want a show of hands about if you had to say right now, what do you want to be? What do you want to do when you grow up? I try to come up with a list of possible careers. Who wants to be a scientist by a show of hands? Who wants to be a medical doctor? Oh, who wants to be a dentist? No dentist. Who wants to be a veterinarian? A teacher. Yay. A lawyer. A business person. An artist of many different types. All right. A politician. A writer. FBI agent, police, or fireman or firewoman. Or the stay-at-home parent. Are there any other careers that I missed? You guys, anything else? Sorry? Shout it out. So a diverse crowd. The biggest vote was medical doctors, so that helps me. Pitch the lecture. Who's female? Who's male? All right. I think a lot of people didn't vote for that. And I wanted to give a pitch that those of you who are interested in science, we need more of you. Science at the moment it's a bit shifted. There are many more men than women earning PhDs, and the demographics of scientists are still very skewed relative to the population. So we need greater participation of people from different ethnic backgrounds, and we need more women in science. So I would encourage you guys to continue to pursue your love of science, those 10 or 15 of you who want to be scientists. So the lecture is going to be divided into a couple of different parts. We'll start with taste, then we will move to smell, then we will have lunch after we've thought about taste and smell. And at the very end, we'll talk about pheromones, which is the most exciting part of all. So a movie was released yesterday. I think it's rated R. But those of you who are allowed to go to R movies can go see it. It's based on a great novel called Perfume, which I highly recommend if you're interested in the sense of smell. I also recommend this book by Chandler Burr called The Emperor of Scent. They're both very affordable. This is a sort of a fiction, nonfiction story of the race for the Nobel Prize in smell. Cell phone? Okay. So let's talk about the sense of taste, why we have it, how does it work, what's it good for. So we have whenever we encounter a piece of food, we evaluate it. You can see that the child eating the candy bar here is getting an enormous amount of pleasure out of it. So the properties are, it's sweet, it's sweet, it's sweet. So in general, animals like things that are sweet. Can someone tell me? We'll do the audience participation stuff now. We'll start the competition now. So why do we and why do most animals like things that are sweet?
Do I press a button?
Just talk.
It's a direct source of carbohydrates.
Go ahead. Say it again.
It's a direct source of carbohydrates.
Excellent. Right. So by tasting it, you can evaluate the nutrient value. So sugar is a carbohydrate and sugar is a nutrient. That's awesome. You guys are good. There are other things like spinach and bitter vegetables that are actually good for us that a lot of people find incredibly disgusting, right? So why, and this is a sort of an innate response. Here's a baby being fed probably some bitter green leafy vegetable, and that's a common response. I don't want to eat it. So why would it be useful to have an aversive response to bitter taste? Any ideas?
Could it be because maybe it helps you if something's poisonous and you don't want to eat it? A lot of poisonous things are bitter.
Excellent. That's exactly right. So we have a sort of an innate aversion to things that are bitter because plants have figured out that they can poison us. Most poisons produced by plants are alkaloids. Most alkaloids are bitter. So it's a sort of a useful thing when your parents tell you to eat your broccoli rabe. It's probably a good idea to be a little skeptical because in the past when our less fortunate ancestors ate bitter plants, they were killed by the bitter alkaloids in them. So we have an innate love for things that are sweet because they tell us about the nutrient value of the food, and we have an innate aversion to things bitter, although we can overcome it. Many people drink beer, which is very bitter. Many people drink coffee, which is extremely bitter. We can learn to love things that are bitter. So, and so the perception of food, of course, is a mixture of taste and smell. That's why I'm talking about taste and smell in the first part of the lecture, because they're really intertwined. When we talk about food tasting good, it's a complete synthesis of the smell of the food and the taste of the food. So actually, these are the only things that we know of that we can taste. So these are the qualities. This is a late addition. So the taste of MSG, the taste of umami, it's a very recent add-on. If you look at textbooks from the 70s, there were only 4 tastes: sour, sweet, bitter, and salt. Those were the only things that were cataloged to be tasteable. We've added a 5th recently, which is MSG, or umami, the Japanese word for yummy. I think scientists out there think that there are more tastes than just this list. Anybody have any suggestions for what else that you know that you can taste that's a different quality from these? It's kind of unfair because I'm controlling who wins here, but...
Spicy.
Spicy is a taste, but technicality. So spicy is tasted by a separate system called the trigeminal, the nociceptive system. So spicy, we know about spicy. It's not technically a taste. And anything else that when you guys eat something that doesn't fall into these categories? When you eat a French fry, what kind of a sensation do you get when you eat a yummy French fry? Salty we have, right? Greasy. Who said that? Who said greasy? Wherever Jennifer is, we'll have her. Is there?
Can something be tasteless? Is that considered another?
Can something be tasteless? So even water has a taste. So even water has a taste. It's difficult. Most things you put in your mouth will elicit some reaction, a texture, a temperature. So even water has a kind of a funny taste to it. So there are scientists who think that that greasiness, that fattiness, is actually a taste. So maybe in 5 years we'll have a molecular definition that will be added to this for fat taste. I think we're pretty close to having that. All right? So science is marching onward and telling us more about how we taste things. So this is an image that's been found in every textbook since the 1880s. How many of you guys have learned that the tongue is divided into different parts? Okay. So it's just complete and absolute rubbish. It's completely wrong. Complete and utter nonsense. Every textbook, your textbook, every textbook that is written still says, yes, at the back of the tongue is where bitter is. Touch things with the tip of the tongue is where sweet is. Complete and utter rubbish. It's been persisting in the literature for over 100 years, and it's completely wrong. In fact, the way things work, here's a beautiful cartoon tongue from Scientific American. The tongue is segmented into different parts. You have different taste buds that have different funny names that are distributed across the tongue. However, each of them has the capacity to taste those 5 elemental tastes. It was a sort of a typo in a textbook 150 years ago that's persisted to this day. And molecular biology in the last 5 or 10 years has completely swept away that illusion. So I'm impressing upon you guys, those of you who will become teachers, to educate your peers and get the textbooks changed, because it's complete rubbish that the tongue is segmented into different areas. Every part of the tongue can taste everything. Why would that be a useful thing, to have every part of the tongue be able to taste everything? Team North?
If part of the tongue is
Go ahead.
If part of the tongue is damaged, you'd still be able to taste with the other portion of your tongue.
So that's right. So if you, it would be bad if you had only the tip of your tongue that could taste sweet, and you had a piercing accident or something that caused you to lose the front of your tongue. The other thing is that we talked about, you mentioned that bitter is used in food rejection or food acceptance, and if you had to wait until the deadly mushroom were at the back of your tongue, it would be too late and would not be an adaptive, intelligent thing for an animal to do. So this is actually the map. If we take the tongue, this is the way the textbook should read now. This is from a Nature Insight review from Charles Zucker. So this is the way the tongue really works. Every part of the tongue tastes bitter, salt, sweet, umami, and sour. And the tongue is organized into these beautiful little taste buds, each of which have all the cells that allow you to taste the different yummy things that make up a taste. And now we have this cool movie with Todd's tongue showing you how taste. We're zooming in on Todd's tongue. We're zooming in on one of these papillae, which have a taste bud in it. And here are all of the different sensory neurons that light up in response to the different tastes that come in. So umami lights up a different set of cells than. So here's our Chinese bitter melon, one of the bitterest and tastiest vegetables out there. It lands on the tip of your tongue and it excites only the bitter neurons. Shake some salt onto your tongue and you get that dribbling across this taste bud, and only the salt neurons fire. You then chew on a sweet candy and you activate only the sweet neurons, and then you have some yummy egg foo young full of MSG, and the umami neurons are lit up. And then you chew on a lemon, and only the sour neurons light up. And then when you chew on sweet and sour items, you have lighting up without any interference the sweet and sour elements. So each of these things work separately. They're separate input channels. There are neurons dedicated to sweet, sour, bitter, umami, and salty, with no crosstalk whatsoever. So we know a lot about the sense of taste from our own experiences of tasting things, but science can only go so far if we only use our own experience, if we only use humans. So I'll talk to you today about a couple different animal models that we can use to get into basic questions for how taste and smell work and how pheromones are processed. For those of you guys arriving late, you are members of the South team, and every time you answer a question, Jennifer will be keeping score. So please participate. So Charles Zucker at UCSD pioneered about 10 years ago using the mouse as a model for the molecular biology of taste. We have all of this phenomenology that we can taste bitter things, we can taste sweet things, but how does it really work? What are the proteins involved that allow us to detect these different items? And so the first thing that you do when you work in a mouse model is you have to be sure that these animals are actually able to participate in your tests and that they have these sensations. So we have to come up with an assay that tells us that the animal has the same reactions to these tastings as we do. We have a South question.
Yeah, this is going back a little bit.
Can we get this one on, please?
This is going back a little bit, but when you were showing us the human tongue, I noticed that an area in the center of the tongue was not lit up for any of the tastes. Is there an adaptive, benefit to not being able to taste anything in the center of the tongue, or can we actually taste stuff there?
I think that it's just the distribution of the taste buds, that there's a region there that's just devoid of taste buds. And I wish I could give you an explanation for why that bit is devoid, but I can't. So it could be that part's used for chewing and you want to I don't really have an explanation, but you're right that there's a non-uniform distribution. Most of the action is at the front of the tongue, which relates to the question of when you have the food in your mouth, it's the do or die moment. You want to make the assignment early, whether you're going to spit this out or not. So that'd be my kind of made-up answer, that the front of the tongue is where most of the papillae are, where most of the discrimination gets done. Thank you, Todd, for your tongue. So we need to figure out whether mice can taste. How do you ask a mouse whether it can taste something? You guys have... Sorry? Okay, let me get you the microphone. We don't have to go through the whole talk. I like answering the questions. Good.
Why are there 3 different types of papillae? Or, like, if they can all do the same thing, like, why are there 3 different types?
They differ a little bit in the numbers of cells expressing the different types of receptors. It's a kind of it's a developmental way that the tongue evolves. They appear to have they appear to encode the same basic functions. It may just be the way that you structure the tongue, that you have things that are morphologically different at the front and the back. But there's no deep functional difference between those different classes of papillae. Okay? So let's figure out whether mice can taste. So the experiment that was done was to give mice. They're sitting in their cages and they have the choice of 2 bottles. There's a bottle that's filled with water, and then there's a bottle that's filled with water and a tastant. And it's a very simple test. You basically look at the number of times they lick from the bottle that has the taste in it and compare it to the water rate. So if they're really thirsty, they'll drink from the water bottle. If they really like what's in the bottle that has the taste, they'll drink more from that one. So who can tell me whether they like, this is sucrose, who can tell me whether they like sucrose? Sorry? Yep, they like it. So depending on how high the concentration is, so 30 millimolar sucrose, they already like that 2 times more than water. When you get up to 1 molar sucrose, which is very, very sweet indeed, then they like it almost 10 times as much as water. So that's the operational test, that mice, first of all, can tell the difference between sucrose and water. They can do so at a really, really, this is a dilute solution. This is not very sweet. And they like it, just like we do. So they psychophysically, they feel the same way about sugar that humans do. So that's useful. Then you do the same thing with glutamate, which is monosodium, which is MSG, the yummy cooking ingredient, or umami. So do mice like glutamate, or do they not like glutamate? They love it. So, and we didn't go over why it's adaptive. Why would these guys like glutamate? Glutamate's an amino acid. Sir?
Well, glutamate is the most common neurotransmitter in the brain, and when you take something like glutamate, it simulates brain function to like want more of that food.
That's an interesting hypothesis. I think that explains why a lot of people get headaches if they eat a lot of MSG, because I think you get a little bit of overwash of glutamate. The other explanation, which I was kind of looking for, is that it's an amino acid. An amino acid is another readout of how nutritive it is. So these are amino acid responsive cells. They like glutamate, but they like other amino acids also. So it's another way to read out whether or not food is going to be yummy, whether it's going to have something good in it for you to eat. And then finally, quinine is a bitter substance. This is the primary ingredient in tonic water, which is a very bitter substance used. It's an old treatment for malaria. Very bitter. So do mice like quinine? Nope. So they will, this is where they choose water. Once you get up to, and the concentrations are really low, here's 10 millimolar. It's 100-fold less than the sugar that we were using. They really don't drink it at all. Once you get up to this, lick rate is 0. Once they figure out that there's quinine in there, they don't drink it. So it's useful. So mice will, give me 1 second. So mice are just like humans. They love MSG. We love MSG. They love sucrose. We love sucrose, and they really hate bitter stuff. Sir?
Does taste change with age?
Taste changes with age. it changes less than the other senses. If I had to order which sense goes first, the sense of smell goes first. It sort of declines at age 40. Then there's a sort of a tie between vision and hearing, which also go. Taste goes a little bit more slowly. Because I'm trying to make the argument that taste is pretty simple. You identify which of these 5 flavors are present. So it's a simpler processing problem than the other senses. So good. So mice and rats are able to perform in behavioral tests. We can tell that they love sweet things, that they don't like bitter things. And this led Charles Zucker to zero in on the tongue and attempt to identify the genes that underlie the sense of taste. And he did this by basically just taking out the tongue and trying to clone genes that were only expressed in the tongue, right? It's a simple hypothesis. You taste with your tongue. Genes that are expressed in the tongue that allow you to taste things should be specifically expressed in the tongue and not in the liver. So it was a pretty simple test. And he identified these different gene families. I'm just drawing the cartoons of what the proteins look like. They're all membrane proteins, so they all sit in the membrane. And they are, with one exception, they're all complexes of 2 proteins. So if you look in very, very close on your tongue, there's a complex of 2 proteins called T1R2 and T1R3 that allow your tongue to taste sweet. And one of these subunits is recycled in the sense of umami. So T1R3 reemerges, combines with another gene called T1R1 to give you the sensation of umami. And then there's a different class of membrane protein called the T2Rs. T stands for taste. R stands for receptors. So this is the first taste receptor, the second taste receptor. And you can completely account for an explanation for how we can taste bitter substances with these proteins. So that was a pretty exciting discovery. We now know the proteins that allow us to bind to quinine or Chinese bitter melon and discriminate it from sucrose. And then in a paper published just in the last month, we have another group of 2 different membrane proteins that allow us to taste sour. So these are proteins that somehow, it's not clear yet, interact with sour substances, weak acids, and allow the neuron to sense that the acid is there. That was another big misnomer in the taste literature, right? We've all, at least until last year, everybody said, okay, there are probably specific receptors for MSG and sweet and bitter, but sour is really simple. Sour is just, it's proton, it's hydrogen atoms, protonated H+, which is sensed by some ion channel, and that's wrong. It's completely wrong. There's a specific receptor that together allows these guys to taste sour. And then another misnomer is that the taste of salt is just a sodium channel, so something that can recognize sodium ions. And that is probably also wrong. So those were pretty hypotheses, just like the tongue map was a cool hypothesis, but completely not supported by experimental data. So we have different classes of proteins that allow us to taste different substances, and they are all expressed in different cells. So here we're zooming in on a picture, on a section of the mouse tongue. And in red, we're labeling the bitter cells. So all of these guys are cells that are expressing the bitter receptors. And in green, we're labeling the sweet receptors. These are just 2 different areas of the mouse tongue. And you can see, the way to look at these pictures is that there are never any yellow cells. You either choose to make a bitter receptor or a sweet receptor. So why would the mouse segregate, as I implied in that movie? Why would a mouse put sweet in one cell and bitter in the other? You could save real estate and put them in the same cell. Why do that?
Because then it's easier to activate one cell or the other. You wouldn't want to activate both the sweet and the bitter cell with one food because they have 2 different reactions.
Yep. So that's exactly what Charles Zucker believes is going on. That sweet is nice. When you eat a candy bar, you're happy and it's a nice thing. Bitter is really, really a terrible thing. There are things like DNA polymerase, RNA polymerase inhibitors, poisons are usually bitter, and you want to have a really unambiguous signal. You don't want to know how much there is. If it's mixed with any sweet, you just want to know, is the bitter there? And the best way to do that is to segregate each of these tastes into a different cell that does a different thing. So that there's no confusion, so you're not confusing bitter from other substances. So this is an experiment which I'll go through, done in the mouse, which is the validation of the cartoons I showed you. I showed you cartoons in this slide. I showed you cartoons of different proteins that allow you to detect different tastes. And now I'm going to show you the data that goes into those cartoons, showing that different cells expressing different receptors are tuned to different taste notes, so that the tongue and the brain can completely figure out what's on the tongue. So, and these are the data. And these are chorda tympani recordings. So you record the bundle of nerves that leaves the tongue. So there's a sort of a cable of nerves that hooks up into every one of those taste buds in the tongue and relays the taste information to the brain. And you can kind of record everything that's going on with the tongue. So if you take a wild-type mouse and you're listening into that nerve bundle and you stimulate it with MSG, the nerve bundle goes. The tongue has tasted umami and you get a little potential, a little neural potential. You put on sweet, you get a potential bitter, sour, salty. You notice that these things have different heights. Different senses different taste senses will elicit different levels of activation, but you can use these recordings to monitor what is the tongue doing. And so what the Zucker lab at UCSD did was say, well, we think that T1R1 is the essential subunit of the umami receptor, so they made a mouse that didn't have it. And so if they're right, if the hypothesis that T1R1 is the essential subunit for umami, you should be able to mutate the gene, have a mouse that doesn't taste umami, and then you listen into the tongue, and there's absolutely no response to umami. And then the rest of the substances still elicit responses, again, showing that there's no cross-wiring of taste. Taste is segregated from the tongue. You do the same thing with T1R2, which is the yummy sweet receptor, and the tongue can still taste MSG and all the other senses, but can't taste sweet. You take T1R3, which is the subunit that's in common between MSG and sugar, and now these mice can't taste neither MSG nor sweet, but they're still really good at tasting bitter, sour, and salty. And then the last two mutants showed that T2R5 indeed is a bitter taste receptor, so this is one where you actually kill the bitter taste cells. You kill the bitter taste cells with genetic manipulation with this receptor, and now these mice are unable to detect bitter. They're completely insensate to bitter. And then finally, this is really recent data that sour taste is mediated by these proteins. So the take home here, the tongue segregates things into different modalities. There's no crosstalk, and there's a dedicated class of receptors for each of these different senses, which is really cool. All this stuff has been done in the last couple of years. So my lab works on fly taste, and working on flies gives you an additional array of benefits than that which is obtainable in mice and humans. And we know quite a bit now about how flies taste. It's been known for several hundred years that flies, of course, can be made to exhibit taste behaviors. You've seen flies flying around your house. They will land on food. Both house flies and fruit flies will select food. And you can, even if you look closely, you can watch them eating it. So in order to, again, figure out whether flies are a useful model, we have to figure out what is their taste universe. Do they taste like we taste? And we know, of course, that fruit fly Drosophilas, based on their name, they love fruit. So if you put out anything fermenting like bananas or beer or wine, they'll come land on it. And the business end of how the fly tastes and smells, taste is located in the labial palp here. So we zoom in on this part of the fly's face. This essentially is the fly tongue, covered with hundreds of little tiny hairs that have different sizes and shapes, and it turns out different types of tastes. So we'll do a demonstration in a little while that shows that humans can segregate tastes perfectly with no crosstalk, mice segregate taste perfectly with no crosstalk, and flies do the same. So the assays for fly taste are really simple and elegant. So you take a fly. Here's a little fly being held by a pair of forceps very gently, and you put a drop of sugar on its feet. The amazing thing that insects can do that we can't imagine, we only taste with our tongue. There's no other part of the body that you can drape over something and get a taste, but insects are loaded with taste cells. So they can, as they're walking on the ground, they get taste information. They have taste cells on their wings. They even have taste cells on their butt. So the females have a taste cell on their ovipositor. So insects have an unbelievable range of sensation that we can only imagine. So you take a drop of sugar and bring it up to a fly's leg, it's tasting it, and it will extend its proboscis because it's getting a taste treat. So that's a very simple assay. You can figure out whether a fly can respond to a taste. Another assay, which is really fun, which we've done with high school students in my lab, is that you set up a sort of a cafeteria tray with sugar and bitter substances colored with different colors. You let the flies eat, and then you look at the color of their belly. So if they like the red juice, they'll have a red stomach. If they didn't, they will have a blue stomach. And if they couldn't tell the difference between them, they'll have a purple stomach. So you can use these kinds of assays to go through and look very quickly at different genetic models of fly taste. And then the last test is you really just watch them behave. You put flies into a chamber and you give them a choice of 4 boxes. This is a square that has sucrose in it. This is a square that has sucrose. These have just agar. And this seems to be a universal thing. Animals like sugar. It's really quite clear. The fruit flies have very little in common between mice and humans in many other strategies in their life, but they will spend most of their time hanging out on sucrose. So, and you can do these sorts of tests, and you can figure out that flies love all sorts of different sugars. They love all of these different sugars, and they really avoid bitter tastings. So again, seems to be conserved in evolution that flies will avoid bitter tastings like quinine, berberine, denatonium, all of these things, many of which are highly toxic. So even flies have figured out, probably through a process of natural selection, bitter is bad. Avoid it. And through recent molecular biology advances, we actually know the names of the receptors that mediate bitter and sweet taste in flies. These look different than the mouse receptors, but have exactly the same function. So a family of genes here encodes bitter taste receptors, and a protein called GR5A is a sweet taste receptor. So again, flies, humans, and mice segregate good and bad tastes and evolve different classes of receptors to test them. And here's a functional experiment that shows that just as when you record from the back of a mouse's tongue and record impulses, that they're segregating the bitter information from the sweet information, that flies do exactly the same thing. So this is a picture from Kristen Scott's lab. So you take a fly and you take a little piece of the cuticle away from the top of its brain, and the brain is expressing a fluorescent protein called Gcamp. So you can watch the fly's brain think about different things. We use this technique to watch a fly's brain think about smells. You can use this technique to watch a fly think about tastes. And you can just bring a microscope down. The fly is alive during this procedure. And you bring a microscope down and you just watch the fly as it's being presented with tastes. So you put a little sugar on its tongue or a little bitter caffeine on its tongue. And the result is as follows. So when you put this activity tracer in both the bitter cells and the sweet cells, and you put on a bitter substance, you light up only the bitter cells. You light up only the sweet cells with a sweet substance, and there's absolutely no crosstalk. So in the brain, flies, just like mice, are segregating bitter from sweet. So it's exactly the same logic, exactly the same organization. There's a question up there. Go for it. Stand up.
One of my former students has been working with mouse. One of my former students has been working with mouse pups with sensory organs at Carnegie Mellon, and I was just wondering with flies if you actually used microfine wires or how you actually measured the impulses, because it's so incredibly small. How are these recordings actually made of the actual activity?
The magic of the microscope. So flies are very, very small. And so flies are, I don't remember what the scale bar is, but you simply, you bring them underneath an objective where you can focus in on the part of the brain. You really are just focusing on that part of the brain. This is the sweet part of the brain, and this is the bitter part of the brain right here. And you then record a movie this is a dye that measures calcium. When neurons are excited, they'll release calcium. And so you can make a movie in time as the neurons are excited, they'll glow. Well, you're seeing the release of calcium by neurons that are getting excited by bitter or sweet, and then you can make a movie of it. And I'm taking some frames from the movie that show that this part of the brain is excited by bitter tastings, and this part of the brain is excited by sweet tastings. You can also put a needle, by the way. You can put a recording electrode here into the fly tongue and measure nerve impulses directly. That's just a more difficult technique. More difficult technique because you have to kind of hunt around and figure out where you're recording. Here fly genetics is used to, we know we're looking at the sweet cells because we're looking at the sweet receptor, right? Does that make sense? Microscopes, useful tools. Okay, so I think this is the end of my pitch about taste. Taste gives you important information about potentially bitter poisons, the nutritive things that can be found in amino acids and sugars, sour tastings, and we segregate the information, whether you're a fly, a mouse, or a human, into different parts that are unambiguous. And you shouldn't take my word for it. We're going to get 6 people up from the audience. I need 3 people from the North team and 3 people from the South team. I will take you, I will take you, and I will take you. Yellow. You, and you. Okay, so I'm going to set these guys up, and then you guys can ask me regular questions. All right. Sal?
Thank you.
All right. I want you to dip your finger into, you can grab a piece of paper. Are you going to south? South, south. So you're going to dip your finger into the bowl and then write down what you taste. And it's color-coded. Each of you has a bowl. All right. Can you find the bowl and write, what does it taste like to you? All right, so while they're doing that, are there any general questions about what I said? Any questions at all? Okay, wait a second.
Sorry, hello.
Yes.
I'm allergic to MSG, and I was just wondering why so many people are.
Describe your symptoms.
Well, I get a rash.
You get a rash.
Yeah.
Now, I'm not a clinician, so I don't think I can really. A lot of people report headaches. I get a really, really severe headache whenever I eat MSG, and I think it's the glutamate. I think it's the glutamate hypothesis, which maybe I'm just making that up. The glutamate. MSG has glutamate, which, as one of your colleagues said, is the major neurotransmitter in the brain, probably going through the blood-brain barrier, making my brain excited, jumbled up, and headachy. I have not heard the rash hypothesis, and I don't have any explanation, except don't eat the stuff, right? It really is just a flavor enhancer, and I would stay away from it. Yes? I have one over here. We'll take one more. Are you guys done?
Is there a reason why there are more receptors for the bitter than the sweet for the fly?
Ah, good question. This is a really good question, and I want you to answer it. Go back. So, and this is the same thing is true in the mouse. So, for the mouse, there's one sweet receptor, T2R. It binds to all things that are sweet. To every sugar. And for bitter, there's probably 80 different types of receptors for bitter substances. Why? And same thing for the fly. So why would that be?
Is it because bitter, it's more, they want to notice it more quicker?
Quicker's part of it, but what would you get from having 80 bitter receptors versus one sweet receptor?
You could get a faster reaction. And
what else would you get, though? What would having more of them? Go ahead. Do you have an idea?
You'd be able to recognize it, even like the smallest amount of bitterness.
That's true. That's a version of that. All these things are right, but there's a real answer which I'm looking for. What would you get from? And there are 80 different ones. There are 80 different bitter receptors. I think that the taste team is done, so we'll get to you in a minute.
Would it be because of variety?
Yes, so if you have 80 bitter receptors, you can have a much broader range of bitter things. Let's give the guy applause. Yay! You have much better power of discriminating bitter taste. You want to have as many, you want to taste as many bitter things as plants can come up with, whereas there's not that many sugars out there, so it will keep you alive. All right. Okay. What did you guys have for orange? Salty. Can I have my microphone? Okay. Can you just hold the question? Orange was?
Salty.
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 Wait for the microphone.
Salt. Salt.
How about green?
Sweet. Sugar.
Sweet.
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 See, complete agreement. There was no wavering, 100% right. They all got it right. And that is what taste is. You can completely figure out whether something, and you'll see how different it is for smell in a minute. Thank you, you guys. All right.
Thank you.
All right. You ready for smell? So smell is it's an entirely different creature, we'll have questions at the end for anybody who has something they're dying to ask me. Smell is an entirely different matter than taste, and it relates to our colleague's answer down here. You want to be able to, more receptors, it can smell more things. So why do we have a sense of smell, and what is it good for? We talked about sugar being good to evaluate nutritional status of food, bitter being good to avoid potential toxins. Sense of smell is one of the oldest senses we have and allows us to sense bad things in the environment, like the smell of a fire. We can evaluate the food by smelling it. So most of what you eat for lunch you evaluate not by tasting, but by smelling it. Animals use their sense of smell to learn things about their mates, which we'll talk about in the last part of the talk after lunch. And if you're a little furry animal, you get really good at recognizing the smell of your predator's urine. So the sense of smell is really key for survival. It's something that you can plan ahead for. Smell acts at a distance. Taste acts when it hits your tongue. So that's why smell is argued to be slightly more important than, also because I work on smell. I think smell is more important. So you can get information from the environment and make decisions like avoid predators and get to food. We still use our sense of smell even though we're highly adapted and we're a really visual species. Every source of natural cooking gas used in the world has this compound added to it so that if there is a leak, a gas leak, you can detect it. Gas is odorless, right? But we add this substance, this sulfurous substance, so we can smell it. That's something that most people can smell this substance, and it's useful to detect gas leaks. And then we also still use our sense of smell when we want to evaluate whether something is still good. You look at the date, but if the date is close to today's date, you're not going to taste it, right? Usually the first thing you do is smell it. And there's an odor, there's a certain odor of sour milk that we're all really good at detecting and usually get rid of it. So we can use our sense of smell to evaluate food quality long before we put the sour milk on our tongue, right? And as I was saying, most of the beautiful, yummy things that go with the taste, the flavor of food have nothing at all to do with taste. So we know that French fries, you'll get that fat taste sensation, you'll get the salty taste sensation, but the richness of it, what makes a French fry, is your sense of smell. It's also it's important to us. Who wore deodorant today? I put deodorant on. Who put deodorant on today? You guys are among the people who masked your natural human scent with antiperspirant or deodorants. We also spend an enormous amount of money on perfume, right? It's a huge industry, $17 billion. And many of these perfumes have really nasty, skunky odors built into them. So humans are weird. We have a natural funky smell, and we get rid of it by showering and wearing deodorants, and then we douse ourselves with perfumes that are full of animal musk. So humans are a little bit weird that way. So we get rid of the chemical communication that might communicate stuff, and we coat ourselves with a synthetic scent. which we'll get to at the end. And then, of course, the huge spa industry. We really are in an age where sensory enjoyment is at a peak in this country, I would say. We spend billions and billions of dollars. So how does the sense of smell work? So we talked about taste being mediated by the tongue. The sense of smell is mediated by millions of neurons in the olfactory epithelium, which resides in the roof of your nasal cavity. And these are millions of neurons whose job it is to detect an unknown number of different odors. So sugar taste is pretty simple. There are maybe the number of sugars that we can taste, probably numbers in the dozens, if that. And the number of smells that we can detect, it's an unknown number, thousands, hundreds of thousands, nobody really knows. And these communicate information directly to the brain about what the smell is. And so what are odors? Anything that smells has to be volatile. So tastes are, by definition, something that has to contact the tongue. You can't taste things that are floating around in the air. Smell acts at a distance. Smell molecules have to go up into the air. And it's an interesting unsolved problem that we don't know. We couldn't look at something and predict what it would taste like or smell like, for that matter. So you can't look at this and say that this is actually a really nasty, rotten egg smelling compound, or that this smells a little bit like bitter almond. It's, at the moment, a completely empirical science. We don't understand what the relationship between a chemical structure is and what it will smell like. And this is a sort of the book, The Emperor of Scent, is about that problem. It's a problem we all acknowledge, that no matter how much chemistry or modeling you put to the problem, we can't make a chemical and then predict this will smell like grass or this will smell like fecal matter. We just can't guess. But how is this accomplished? The nose can do amazing things. So you can take two chemical isomers that have exactly the same formula weight but are left and right isomers, and they smell different. So the nose is somehow able to tell the difference between this kind of the plus carbon and the minus carbon, and they have completely different sensations. So there must be neurons that can tell them apart, and then they send different signals to the brain. So that's pretty cool. And as I was saying, these things both smell like bitter almond, but they have very different structures. So you can have things that smell the same, look different, look the same, smell different. So the rules of how something smells are still mysterious, and it would be useful to solve that. We could make very beautiful perfumes if we could plan them rationally. So the big discovery from my point of view, because I trained with Richard Axel, is in 1991, he and his postdoc Linda Buck figured out what are the molecular receptors that allow the nose to do this? How can we smell hundreds of thousands of different molecules and tell them apart and get conscious percepts of them? It's a family of proteins, again, membrane proteins, just like the taste receptors. They sit in the membrane of neurons in our nose, and there are thousands of them. So if you're a mouse or a dog, you have between 1,000 and 2,000 different odorant receptors. We were talking earlier about having 80 bitter taste receptors. Gives you a chance to smell more, taste more things. Imagine being a rat or a dog with between 1,500 and 2,000 different receptors. So this is the power of the sense of smell. There's a diversity of receptors. We're a little pathetic. We have 350 functional odorant receptors. But still, the mathematically inclined in the audience, if I give you that any one of those 350 receptors could bind to 20 to 30 different smells in different combinations, the combinatorial of the different kinds of things you could smell is huge. And so for this, these guys got the Nobel Prize in 2004, which was great for the field because who cares about smell? But it was great in 2004. We got all this publicity and people are flooding in and doing a lot of work, and it's a very exciting time to work in smell. And the big insight into this problem of how does a nose smell 100,000 different molecules was solved by the cloning of this gene family, which Linda cloned, using the same logic that Charles Zucker used. So where would you, if you were looking for odorant receptors, this unsolved problem, for hundreds and hundreds of years, nobody knew how smell worked. Where would you look for the smell receptors? Get the microphone out. If you wanted to clone the odorant receptors, which were not known, where would you look for them? This is a really stupid question with a simple answer. Up there. Go ahead. The nose, yes. Yay. No, but it's a really, that was the big advance. So that Linda Buck looked for it in the nose. What else would you look for if you were looking for odorant receptors? The idea was that you can smell 100,000 things. What would you be looking for? I'll wait all day for an answer. Come on, you guys. You want to be able to smell 100,000 things. Are you looking for 2 proteins? What are you looking for? Oh, pink shirt.
For 100,000 different proteins.
Exactly. You're looking for a lot of them. You're looking for a lot of them. If your model, if your hypothesis is that for every smell there's a receptor, you're looking for 100,000 proteins. If your hypothesis is there may be some crosstalk, maybe you're looking for 1,000. And that's what Linda looked for, and that's what she found. And what she also found is that here's a model of the protein as it snakes through the membrane. And all the residues in pink are hypervariable. So all of these amino acids are different across the family. And so the hypothesis is that is where the smells are binding. That that's the pocket where the proteins are able to tell the difference between cinnamon and nutmeg. That there's a few little amino acid changes in receptor 22 that is different from receptor 23 that allows this protein to tell the difference between nutmeg and cinnamon. And so that is what they solved in 1991, and that's why they went to Sweden and cashiered $1 million. What happens in the nose is that the odorant binds this receptor, the buccal receptor, and that initiates a cascade of events that gets the neuron to be activated, which we will, which brings us to the problem of how do you then do it? You have a nose that has millions of neurons expressing hundreds or thousands of receptors. And the problem is that if you smell a rose or these beautiful lilies, if we were to get a chemist in here to pull all the smells off the lilies, there are probably 500 molecules coming off the lilies, many of which are shared by the rose, but differ in the proportions of which smell molecules are there, and the nose has to put that together. A lily always smells like a lily, smells different than a rose. Roses have over 260 molecules, and so the job that the nose has to do is to put that together, for which we have this other really cool movie. So floating around in the air are these smell molecules, and then they interact with sensory neurons in the nose. These are cartoons of sensory neurons. You have red neurons, yellow neurons, green neurons, blue neurons. And unlike the tongue, where there was a one-to-one match, what happens in the nose is totally different. If you have the blue odorant coming in, it will bind to the yellow receptor a little bit and activate the neuron. It won't be able to fit into the green receptor, but it binds the red receptor perfectly. It's a perfect match. And the red neuron is activated a lot. The yellow neuron's activated a little bit. The blue neuron's activated a little bit more. And the yellow neuron is not activated at all. But there is then this combinatorial signal that comes into the brain that allows, in our case, 350 receptors to be able to smell 100,000 compounds, because we have this beautiful crosstalk. A given odorant will activate 10, 20, 30 different groups of receptors, and through that combination, you're able to get the richness of the sense of smell. And so that's the problem the brain has to solve. We don't understand at all how that happens, how that works, and that's the big frontier. Those of you interested in the neurobiology of the brain, how do you get a simple sensory epithelium in the nose to be able to take in, even to sniff the lily, get 500 different molecules in different concentrations and put that together? It's a huge neurobiology problem. Odors can be, just like sugar can be inherently pleasant and bitter can be inherently unpleasant, we also have smells can be inherently pleasant or very unpleasant. But a lot of this is much more dependent on your point of view or your cultural background. So I picked 6 people at random. They agreed 100% on this is sugar, this is sweet, and this is salty. People will disagree enormously on what is considered unpleasant or pleasant, depending on their cultural background, their genome, if they have missing odorant receptors. There's a lot more variability in the sense of smell, which plays out in something called a specific anosmia, which we study here in the Rockefeller University Hospital. We collect people that have these interesting anomalies, not the MSG allergy, which is a different kind of a thing, but the specific anosmia where someone will come in and be able to smell everything, but will not at all be able to smell vanilla, or this musk, or the smell of sweaty socks. And we're trying to figure out how do you get these people? If the sense of smell is so combinatorial, what is the exact genetic defect in these people that allows them to drop out one part of their odor universe? Here's some other fun facts. As I was saying before, sense of taste lasts longer than the sense of smell. At around my age, it begins to kind of begins to be over. You begin to discriminate less. You become less sensitive. Women as a whole are more sensitive than men. I think that explains a lot of the discord between the sexes, right? That there's women in general find most odors much stronger and more unpleasant than men. So there will be these disagreements in households over whether something smells. Women who are pregnant or in the menstrual cycle will have very, very different. You'll become much more sensitive to smells when you're pregnant and when you're at a certain point in your menstrual cycle. And for all of the doctors, the future doctors in the audience, the sense of smell, I think more and more will be used as a clinical diagnostic tool as an earlier marker for Alzheimer's disease and other neurodegenerative disorders, as well as schizophrenia. There seems to be something about the identification of odors that goes awry in schizophrenics, which I think relates to the problem of the complexity of how the smell map forms in the brain. It's easy to break. And then people who completely lose their sense of smell often have serious problems with depression and eating disorders. And now we're going to do our smell demonstration. So I need 6 different people. We're going to go to the back. Okay, we'll take you. And you, the white shirt. And you. And you. And you. And... Okay, and you on the ends? All right. So you guys are south. Just south. And we'll do north over here. Okay. You guys with me? Yes. You're going to open up the blue vial. We should get away from the flowers because they're very smelly. Open the blue vial, smell it until you figure out what it smells like, and write down what it smells like. Adjectives. If you need help, there's a list here. And then do the same thing with the orange, okay? I'll move this out of the way.
What side are you on?
If you need help, you can choose an adjective from here, or just kind of describe with an adjective. Are there any general questions that came up back there?
Why does your sensitivity of smell change throughout the day?
Sorry?
Why does your sensitivity of smell change throughout the day? Like during the morning, you feel like you don't want to smell a certain thing.
That could just relate to the general problem of what's called the arousal state of the brain, which is a kind of a general thing for everything you do. Like when you're just beginning the day, it's harder for you to get your kind of motor patterns to get your walking going. So I think it's just a matter of how awake the brain is and how awake and how ready the brain is for information.
Hi. Is it true that we never forget a certain smell?
Yeah. So this is so Proust wrote a book about his Madeleine that bring back a flood of memories. Smell is anecdotally said to be the best trigger of memories. I happen to agree with that. I think of the other. So you could see something from the past and you might remember something. You could taste something from the past, maybe even feel something physically, but the sense of smell does seem to have a more anecdotal ability to cause a flood back of memories. Some of that may relate to the way that smell is wired into the brain. It may have a more intimate connection with the memory centers. A lot of it may just be an emotional things that humans feel. I would love to get that set up in the mouse where you could separate all the human emotional issues from this question and get a mouse to do it, which is true in some cases that in flies, the best way to get a fly to remember something is to give it a sense of smell with something it should remember, like an unpleasant shock. So smell seems to be one of the most potent, rewarding cues in animal learning and memory. Anything else?
How come when you're sick, like you have a cold, one of the symptoms is that you don't have a really good sense of taste or smell?
Okay, excellent question. So whenever you have it's typical in the winter that people will completely lose their sense of smell, and with it their sense of taste. And that's because the sense of taste is largely your sense of smell, right? Food doesn't taste good because all you taste is the bitter, the sour, the sweet. So what happens is that any irritation or inflammation of the nasal epithelium will either disturb the chemical balance of the mucus, which is important in the sense of smell, or will actually damage the smell neurons themselves. And so smell neurons regenerate. So if you have a big infection, you'll kill off all of the neurons in your nose, and you'll lose your sense of smell. And then in time, a week, 2 weeks, 3 weeks, the cells will be reborn from stem cells, and you'll get a new sense of smell. So in some cases, it never comes back, and those people are left with a permanent anosmia.
Is it true that, does smell wake you up?
Sorry?
Can smell wake you up?
Can smell make you what?
Wake you up from sleep.
Wake you up, yes, absolutely.
Is it true that
So it's
fat
in
and
a
smell
stimulus.
higher?
I mean, it's like any sensory stimulus. If you're asleep, you can be roused by any of the stimuli. Probably a touch stimulus is the strongest. Let me see how these guys are doing. Are you done with your adjectives? Yeah. Awesome. Awesome. All right. Our contestants are done. This is so fascinating. Okay. Hold on one second. So wait for the microphone. So what did you, we're going to go through the blue odor first. The blue odor was let me see what it was. Yeah. Isoeugenol. What did the blue one smell like?
Vanilla.
A warm, like vanilla sweet smell.
Vanilla.
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 You guys all, this is the problem. Like, if this were a real experiment, they would have been locked in separate rooms. And now, the orange one. Did everybody smell the orange one? Could everybody smell it? Yes? Okay, what did the orange one smell like?
Alcohol.
Alcohol.
A sour nail polish remover smell.
Nail polish remover.
Nail polish remover smell.
Nail polish remover.
I got caraway. I don't know.
Caraway.
Nail polish remover.
All right, thank you. All right, it was a success. All right, that was awesome. So that was a total success from my point. The first odor was isoeugenol, which smells a little like vanilla. Most people know what vanilla smells like. It's really a familiar odor, and I think, although there may have been some cross-checking of notes, everybody thought it smelled like vanilla. The other odor was androstenone, which is a breakdown product of testosterone. So before we shower, all the men in the room would have androstenone on them, and there's a huge variability in how people feel. Some people can't smell it at all. Other people think it smells pleasant. People smell like caraway or nail polish remover. There's an enormous difference in how people perceive that particular sense of smell. All right. So I think I rest my case. So the sense of smell, it's more variable. It's more dependent on the eye of the observer. It's harder to categorize. It can be really hard to categorize. And now we'll put the descriptor sign down. I'm going to spend the next 10 minutes talking about the sense of smell in fruit flies, but I'm going to skip some stuff because you guys are a good audience. You guys are such an awesome audience. You asked me so many questions. We're going to skip something. So we have a paper coming out next week in Nature, which concerns the question I've always been fascinated in. How do mosquitoes find us? How do they find us? We're walking around. How do they find us? What is it about humans that makes us so attractive for the female mosquito? So in the January 4th issue of Nature, we will have a story coming out about carbon dioxide receptors. So that's part of the answer. Every time we go out in the woods, we breathe. And when we breathe, we exhale carbon dioxide. And that's part of the signature that makes us human. Humans smell like humans. If we're camping, we really smell like humans. If we're camping and we haven't showered, we really smell like humans should smell. If we're freshly washed and covered with perfume, we also sort of smell like humans because there's nothing else out there that smells like that. And we release carbon dioxide. So that's the signature that mosquitoes are looking for. They're looking for something that is warm-blooded, so it emits heat. But the most important things are our smell, our host odor, because we're the host, and the carbon dioxide that we exhale in our breath. So this ends up being something that's not unique to mosquitoes. Insects are really, really sensitive to this stuff. The person next to you is exhaling into your face 5% CO2. And aside from the sensation of maybe a funny smell or unrelated to the CO2 or warmth from the breath, humans cannot detect CO2 at anything except extremely high concentrations. If you go out there and pop a can of Diet Coke too close to your mouth, you'll get a stinging sensation. That's your nociceptive trigeminal nerve reporting very high concentrations of CO2, but we can't smell it. Whereas mosquitoes can smell tiny fluctuations in CO2 that helps them hunt us. Other insects also use CO2. So here's a slide of a hawk moth zeroing in on a datura flower. The flower has just opened and it's released a puff of CO2. That tells the moth that this is good eaten, and it flies up to it and drinks from that particular flower. Fruit flies, which is our model, they also detect CO2, and we do this in the lab. So we put a bunch of flies into a tube. We put them into something called a T-maze, which is like an elevator-like contraption. And we then lower the elevator, and the flies are here waiting for something to happen, and then we give them a choice. They can go to the left, where there's carbon dioxide, or they can go to the right, where there's air, which has 350 parts per million carbon dioxide. Flies, unlike mosquitoes who love CO2 and moths who love CO2, flies hate CO2, so they run away. They run away from CO2 as quickly as they can. Very sensitive assay. We can figure out that they can respond to it very, very sensitively. Even if you mix something that they love, like yeast paste or beer or wine, with enough CO2, they will run away from it. We don't understand that, but it's a very good assay. And so here we graph the avoidance index. Between 85 and 100% of the flies avoid it. So this is the question that my student Walt Jones asked. He just recently graduated from the lab. He's in the MD Ph.D. program. So the question that we wanted to ask is which neurons, here's the fly head, how are the flies sensing the CO2, and what's the molecular receptor? So we already knew from previous work that I just skipped over that if you record from a group of neurons here, you put one of these tungsten electrodes that my colleague in the vest asked me about, you put a needle in here, you can record nerve spikes from a fly. And there's a group of neurons here. This one likes ethyl acetate. So we're recording in time. Each of these is a neuron getting excited. You puff on ethyl acetate, and you see that the neuron's getting very excited. You put on acetoine, which smells like artificial buttered popcorn. Flies like that too. The cell gets excited. And here's the neuron that smells CO2. So you put on CO2, the neuron gets visibly excited. Here's methylsalicylate, which is wintergreen. The little neuron gets excited. And so we figured out, and previous work from others, that our neurons here on the fly antenna, which are very, very selectively tuned only to carbon dioxide, a little reminiscent of the sweet taste neurons in your tongue or in the fly tongue. So these neurons do one job. They detect CO2. And previous work from the Axel lab and from John Carlson's lab at Yale showed that the neurons, you can make them glow green by a genetic trick, and these neurons express a receptor called GR21A, gustatory receptor 21A. And there was an experiment published in Nature a couple of years ago showing that while flies avoid carbon dioxide, if you get rid of these neurons genetically, you take the neurons away, the flies now can't tell the difference. So half of them will go to the CO2 and half will go to the air. So we knew that these were the only neurons on the fly antenna that allowed them to sense CO2. And we knew something about that the GR21A, which is sitting here in this big tree of chemosensory receptors in the fly genome, that this is a taste receptor that's actually expressed in the fly nose. So most taste receptors are expressed here in the tongue. Most odorant receptors are expressed here in the two smell organs, but we have one taste receptor expressed in the antenna. And we wanted to know, is GR21A the CO2 receptor? Nobody has any idea in any animal on Earth how do animals sense CO2, and we wanted to go after it. So Wald found that not only does GR21A mark the CO2-sensitive neurons, but there's a sister gene called GR63A, which is always co-expressed with GR21A. So when you look, all of the neurons, here we're looking at a close-up of the fly antenna with our microscope, and we see that we have 1, 2, 3, 4, 5, 6. This is just dirt. We have 6 neurons, and they always express both of these guys. You do a control experiment to show that we can tell the difference between green and purple. In green are the CO2 responsive cells, and in purple something unrelated. So when you get white, it shows that these guys are co-expressed. We then zoom in on the fly brain, the part of the brain. Here we're zooming in. This is the antenna. The neurons send information and neuronal information into the brain. And we knew previously that it was this part of the brain that is selectively activated by CO2. And lo and behold, that part of the brain is marked by the expression of those 2 genes, which mark the CO2 receptor neurons. You see where I'm going to go with this, right? We thought these might be the CO2 receptor. So what Walt did was say, well, if these 2 proteins encode the CO2 receptor, we should be able to move them genetically into a neuron that doesn't care about CO2, that smells only fruit. So you take a fruit neuron. You take normally a fruit neuron in the fly antenna. You puff on fruit odor, and the neuron goes wild with the smell of fruit. You puff on CO2, it does nothing, because the neuron doesn't like CO2, it doesn't care about CO2. What if we were to move these 2 proteins into a fruit odor neuron? What would happen? Would we be able to record from that neuron and figure out if it now likes CO2? And magically, this is exactly what happens. So the neurons are always sort of chattering in the background. There's always a lot of kind of nervous chatter in the nervous system. So the neurons are spiking, but not in an organized way. Here's where you have the puff of CO2. You move this receptor, nothing happens. You move that receptor. 21A alone does nothing. 63 alone does nothing. This combination does nothing. That combination does nothing. But bam, you put 21A and 63A. Now you get a fruit neuron that is wildly excited by CO2. You get a huge CO2 response. And the response looks pretty close to the kind of response that you get in a normal CO2 neuron. This is a normal CO2 neuron. The more CO2 you add, the more interested it gets. Same thing happens with our artificial situation. So the proof of principle would be to figure out whether any of these 3 models is the case. Since we have 2 receptors, it might be that only one of these candidate genes is actually the CO2 receptor, if this is a model for CO2. Or it might be that they work together as a group to make a CO2 receptor. So to test this, Walt used fly genetics to rip the GR63A gene out of the fly genome. So we made mutant flies that don't have that receptor. And then he recorded from those CO2-sensitive neurons, and they no longer responded. So the neurons completely no longer respond to CO2. Complete loss of all CO2 responsiveness. And if you restore the gene, the response comes back. And you see the same thing in the T maze. So if you ask, where do the flies go in this T maze? If you are a mutant and you lack the gene, you you cannot sense CO2. And that says that GR21A and GR63A must be working together as a whole to make a carbon dioxide receptor in insects. But fruit flies are nice sort of laboratory insects. We were really interested and concerned about this insect, so the malaria mosquito, which spreads, it and its brethren in the mosquito family spread a large number of very bad infectious diseases like malaria, dengue, yellow fever, West Nile encephalitis, many insect-borne diseases that kill many millions of people annually, mostly in sub-Saharan Africa. So we wanted to know more about how this animal does it. And so Walt found that the CO2 receptors that we found in Drosophila had closely related relatives in the mosquito. And he found that just like in the fly, they hang out together. So we have the 2 homologs are always co-expressed in the sensory organ, in the maxillary palp, which is the organ that detects CO2 in mosquitoes. So this leaves us with the hypothesis that these 2 guys are co-receptors for carbon dioxide chemosensation, and that by extension, although we haven't shown it, right? I haven't shown you anything about mutant mosquitoes that can't. That would be a useful thing. If we could make a mutant mosquito that couldn't sense CO2, this would be a useful thing. These are our candidates for doing the same thing in the mosquito. And so a large part of what we're doing, which is funded by the Bill and Melinda Gates Foundation, is to make a better mosquito repellent, make a better insect repellent by learning something about insect smell and coming up with chemicals that are safe to spray on your body that work based on a known mechanism. So if you could come up with a way to inhibit the CO2 receptor by adding some formulation to our current insect repellents, this, in principle, should be a rational way to design a new insect repellent. And that's all I'm going to say about smell. So welcome back from lunch. This is the third part of the talk. I'm going to talk about the other part of chemical communication that involved in sexual selection and mate choice, the magical power of pheromones to influence how animals communicate with each other. I won't have any of that kind of audience participation, but we'll have some kind of participation relating to the pheromones. So, this is an example of the mating dance of the fruit fly. So they need to figure out some things. The male and the female approach each other. The male taps the female's abdomen. He'll extend his wing and sing her a song. She'll listen to the song. If she likes it, they'll continue. And the whole thing culminates in mating. So there's a lot of communication that goes back and forth in mate selection, and that's what we'll talk about, how animals communicate to each other, that this is going to be an appropriate partnership. Again, I have some suggested reading. So this is an excellent book, much higher price point, from Tristram Wyatt, all about pheromones and animal behavior. And then this is a book called The Scented Ape, about the uniquely human experience of the smells we produce, and historically how we have dealt with the smells we produce and how we've masked them. The Biology and Culture of Human Odor. These are both really good reads at a higher price point than the previous books, but a lot of fun. And if you need anything answered that I didn't get to today, or you need information about any books, just email me, leslie@rockefeller.edu. So in this part of the talk, I'm going to discuss this whole process of how animals use their many senses to communicate information, with a special focus on chemical communication. So, which gets us to the question of, how do animals look for a perfect mate? What kinds of qualities are you looking for? And I posed this question to my relatives last week, none of whom are scientists, and we came up with a list of 13 things. And I forgot to say that Jennifer's been keeping track, and we're going to give you a midterm report on who's winning, south or north.
So after round one, we've got north with 23 and south with 15. So north's a little ahead, but it's not too late.
It is not too late, and so there are microphones. There's a microphone there on the south side, a microphone there on the north side, and there should be people in the audience with microphones who will come to you. We have a big chance here to redress the imbalance. So just in general, you're an animal, you're looking for a mate. Give me some ideas for what you need to evaluate. Just wait for the microphone and say it.
Health. Whoa, it's not working. Oh, the gate. Health.
Healthy. Excellent.
Intelligence.
Huh. Anything else? How about some really, really basic things? Just basic. Pick anybody. It's your job to pick.
Friendship.
I'm going to put that into these kind of personal. I'm going to put that into a category of compatibility. But remember that I'm talking about anything, like an animal, like I'm talking about partnership of two mice or ferrets. I'm not just talking about humans. What are some basic things that all animals?
Physical appearance.
Strength.
Sorry?
Strength.
Strength. Which relates a little bit to physical appearance. How about
Fertility.
Fertility, excellent.
She's over there. Smell.
Physical ability.
Sorry?
Physical ability.
I'm going to put that under strength. I think that's the same thing as strength. Physical ability is the same thing as strength. It's good. We're starting to. How about some super basic things without which children cannot be produced?
No. Financially stable.
Okay, she's confused though. She keeps looking at the side. So I need to.
I'm putting, because animals don't do finance, so we're going to make it general.
Gender.
Gender.
Loyalty.
We're going to put that under loyalty. Put that up here.
You need a good sense of humor because life is difficult.
But remember, we're not talking about boyfriend, girlfriend. We're talking about just in general if you're an animal. I don't know how to measure sense of humor in animals. What are some basic biological things? I'll put that all up here.
Social status.
Social status.
Not such a big age difference.
Age match, very important. Can I get two more? And that one over there.
Species?
Yes! 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 That was the biggie. That was the biggie. Somewhere over here? Was there another one over here? Okay, she got the points for species up there.
Metabolism. Behavior.
You have to say it again. Behavior. Can you expand on that? How do you mean?
It is compatible.
Compatibility. Same thing as compatibility, right? We need one more. Let's go for one more. Responsibility. Okay, we're getting a lot of human. These are all sort of human things. So responsibility, loyalty. Can I spell it? How about the most basic thing of all that would make you able to mate with someone? What must that creature be? Alive! You guys are awesome. And so you guys, it was pretty good. This is what my family came up with. So, but these are important. Receptive. Okay. So this is a lot for two animals to figure out about each other, right? Is the animal alive? Is it the same species? Is it gender matched? It has to be a different sex from me for sexually reproducing animals. Has to be sexually mature, but still fertile, right? You guys got that. Good genes, that kind of played out in healthy and strong. Receptive, meaning that the other animal has an interest in mating with you, and yet is attractive to you. And we went over a lot of the things that considered not a close relative was a big one we missed, right? Like your sibling might meet all of these criteria, but not a close relative is a really important thing. I know it's goofy, but it's obvious and very important. Geographically accessible, like you might hear an animal from far away that sounds good, but you can't get to him or her. Physical attributes, social attributes. So we got a lot of these. So the question is, do you have a burning question? You have a comment about the list? Can I have a microphone?
Like, what about ability to adapt? Like, wouldn't that be something you...
Ability to adapt.
Yeah, to the environment or...
So, ability to adapt would mean that you would be able to sense that the partner would be able to, in case of impending global warming, that their genes would have potential. I agree that would be a great thing to look for, but I think it's hard to ask up front whether or not someone's gene pool has that capacity. But good genes is a version of that there are good genes, right? All right. So the question is, how are you going to get all this information? You could look at someone. You could use vision to figure out whether all these attributes are in place. You could listen to them. You could smell them. You could taste them. You could use your senses to figure these things out. But you guys did very well. You got I think all the essential things. That's awesome. So the question is, we can use all of our senses to make these decisions, and some things like strength are easy to see. Things like good genes are a little bit harder to see by looking at someone, unless the genes actually do something to advertise that they're present. And then you need to be able to communicate this. If you're an animal, the male needs to somehow call out to the female to begin the conversation. So we'll spend some time, I think, going over, if you were trying to come up with the best way to communicate all those things on the list that we drew up and that you drew up, you'd want to be able to send the signal cheaply, and you'd be able to have it be received cheaply. You want it to be discreet so that whatever you advertise doesn't cause you to be eaten by a predator. Like if you're a mouse and you're using a very flashy advertising strategy that gets you noticed by predators, in the long run, you may mate in the short term, but you'll be eaten in the long run. So you need to communicate discreetly. Ideally, it would work in the dark. It would work around physical barriers. Whatever signal you're sending would last a long time and work over a long range and would be easy for the recipient of the message to find you. So we'll go over all of the kinds of signals that animals, and I have some goofy pictures of how humans use exactly these tricks, because we haven't invented any new tricks that animals haven't thought of. Visual signal. So here's the male peacock. So these guys are awesome. They communicate something. They communicate. What is the male peacock communicating here? Because we drew up the list. South.
That it's male.
Yes. And what else? Anything else? because females don't do this.
Physical
right?
appearance.
Sorry?
Physical appearance. Physical appearance.
Right, so it says something about the physical appearance, because the females can find this beautiful, right? But there's another thing. It's not just they find it beautiful, there's something else built into the message. We need like 50 microphone people. How about there? He's beautiful, but what else is he telling the female?
He's an eligible bachelor.
Right. Anything else? Because somebody over there mentioned behavior,
right,
so he can just listed this behavior up there. Anything else?
Strength.
Strength, because he has to walk around with this ridiculous display, right?
No, I mean like he has many eyes, so he's imposing. Excuse me.
So this is a big advertisement for strength.
Ability.
Ability. Yep. But what are the costs of this? What are the costs of this kind of communication? Is it cheap? It's an expensive thing because you have to, as you're a little bird embryo, you have to grow all these feathers and you need to get enough ATP to keep them going, and it's energetically a very expensive way to communicate. How about as far as safety?
He's alive.
Sure, it also says that I'm gonna get rid of this, but as far as is this a safe display if you're walking around? So there's a problem with safety because it's a very showy display. The female will see it, but so will whatever wants to eat you. So this is a visual display and then I think I have like a silly human picture next, so get ready. So humans also use like insane. So let's not kid ourselves. Humans, we do things to our appearance that advertises our strength, our suitability, our availability. So we're doing exactly what the male peacock does. So now we get to auditory cues. So you've all, although we're all city people, right? You're city kids, I'm a city kid. There are occasionally birds here, and the birds sing, and it's usually the males who sing, and they sing a song. So what are the benefits of using auditory cues? What's good about an auditory cue? About communicating things on the list of 13?
If it's audio, like it could either be, I don't know, I like learn on TV. You could have it could be like long range, like she was saying.
Long range, yes.
And there's also I think ultrasound or something, so it can only be heard throughout the species or something.
Yeah, so some animals can communicate in ultrasound, so it's safe, but birds don't, right? So everybody can hear the birds singing. How about, is it cheap or expensive to communicate like this? You have to sing and sing and sing and sing and sing and sing until you get the mate. So I'd argue and then the more extreme case is the cricket. Couldn't get the audio clips, but crickets will basically from dusk to dawn will sit on a tree and rub their I can't remember the body part that generates the cricket's trill, but they sit there for basically 8 hours and trill. So energetically, very, very expensive. How about the safety? What's the safety angle for acoustic communication?
Well, it's not that safe because your predators can hear you too.
Exactly. So it's not discrete because it's not in a way the bird song is very specific because birds have songs that are unique to the species. So it will communicate part of the message. It will say I am this species and it helps them get together. but the predators can also find you, and that's a particular problem for the crickets because all animals are very good at localizing where a sound comes from. So we can, if you drop something on the floor with pretty good precision, if you have both ears functioning, you can find exactly where it fell because we're all very good at figuring out where a sound came from. So auditory cues have some utility, but they tend to end up being expensive to produce and not very covert, so you can open yourself up. And humans, of course, also, we love our rock stars, and we find the songs of the rock stars unbelievably alluring. These are, because I'm very old, I have all the old rock stars up, but Iggy. So I'm going to sell you on this idea that chemical cues end up fulfilling more of the criteria on that little list of being easy to produce and very specific. So chemists for years were looking for a cockroach sex pheromone, which sounds really gross, but it's useful. If you want to improve the performance of your combat bait trays, the thing you want to put in there is the cockroach sex pheromone, and then you draw in every single male roach in Brooklyn goes into your trap, right? So they found it. They found the chemical. This is what the female cockroaches secrete, and it drives the male cockroaches wild. They will run to the source. So it's a cue for there is a female present, and it has these kinds of benefits. This is unique to cockroaches. Only the females produce it, and it sends a signal to the male cockroach that there's a female. She can lay the trail. The male can follow it. And only cockroaches can pick up on it. If I were to have a vial of cockroach sex pheromone, I don't. If I had it, nobody in the room would be able to detect anything because it's a really specific signal. And humans also, of course, I'm going to argue throughout the lecture that we've missed this opportunity, that humans, that we're constantly secreting pheromonal cues, but our culture has advanced to the point where we really, by and large, don't use the chemical cues anymore because we bathe and we wear a lot of perfume and we use visual selection to choose. We have a much more complicated approach to mating, as many of the things on the list there are completely foreign to animals. Friendship, all these important things for humans, friendship, loyalty, completely alien to animals, right? So we've come to a point in our development where we use different kinds of cues. And so now I'm going to talk to you about different kinds of pheromones and how they work in action, with a really cool movie of the silk moth, Bombyx mori. So these are animals that live in the dark, so they're nocturnal. So for most moths, a fancy visual display is useless, right? Because you're awake at night, and so a visual display is useless. You need a chemical display. So females will release this substance that immediately kicks off a mating ritual of the male, which is shown in this clip from Japanese TV. Here are 5 male bombyx, and here is a female. And they were doing nothing until the glass was lifted from the female, and then instantly they figured out that the female was there. They go toward her, and they mate. So it elicits this immediate behavioral reaction, an immediate reaction that says, there's a female, she's alive, she's a Bombyx mori, and she's over there. So it's a very, sorry? It's rapid. It was a very rapid. And you can do this, you can play tricks on these animals. So here's a male moth, here's a bit of female pheromone, and he has detected it and is flying toward her and is initiating a mating ritual. So for insects who live a very short life, they don't have 30 years to find a girlfriend, right? They can't be choosy. They have 2 nights to find a girlfriend. And so chemical communication ends up being the only way to go. You live at night, you sleep during the day, and so to have a system where the female releases a pheromone and the male has evolved to detect it is the most efficient way to go. There was a paper recently in Nature a couple of years ago that showed that mammals also have pheromones. I love teaching this paper to our medical students and our graduate students. I'm going to run it by you. So these are rabbits. It's a special breed of rabbit. These are little baby rabbit pups. And the mothers are very bad mothers, if we look at their behavior. So the mother leaves the pups in the burrow for basically 24 hours, minus 10 minutes. She'll come in for 10 minutes a day, and nurse them. So they have 10 minutes to get all of the food that they're going to get in that 24-hour period, and she ignores them the rest of the time. It looks like really neglectful mothering. In actuality, these are very tasty tidbits for predators. The predators learn to follow the mother, go into the burrow and eat the babies, right? So she kind of avoids hanging out with her children to protect them. So they have to figure out, mom's going to come. You know, we're waiting for 23 hours and 50 minutes. Mom is going to come. We need to eat. So they need to be awake, and they need to figure out that they're ready to eat. And so scientists went in and said, there must be a pheromone that, because they're blind, right? They can't see. They're blind little rabbit pups. They must have some way of knowing that mom is coming. And so they looked for a pheromone. They found it. 2-methylbutyrenal is enough. This little chemical is enough. You put it on a glass rod. The little rabbit puff sniffs it, and it opens its mouth and tries to suckle on the tip of glass. So they found the pheromone. Just that, a little drop of that odor, 2-MB2, is enough to elicit this behavior. So where did the scientists, they had this hypothesis. There must be some chemical cue that tells the babies that mom is coming. Where did they look for it? Where would you look for it if you were trying to look for a pheromone that controlled this kind of suckling behavior in these rabbit pups? Can you use the microphone?
The mom's teats, I guess. Like,
Uh-huh. And can you be more specific?
Okay, so you see how the baby triggered that reaction on the baby? Like they opened its mouth, so I'm guessing that it's used to having that scent or the pheromones coming from where it nurses, like where it suckles.
Okay, so I think that's a great suggestion. So they started there, and they looked. They sort of swabbed the area around where the milk comes out, and they didn't find anything. So where else should they look? Any other ideas? That was a great place to start, and they started there.
I guess the skin, because like that's the first thing you'll probably smell, because that's the first thing that you see.
So they looked at the skin, and they didn't find any evidence there was anything active. I mean, they could have given up and said, whatever, it's not a chemical. They sort of bathed different parts of the mother rabbit, and they couldn't find anything that elicited that suckling behavior. So where else did they look?
The mouth, maybe when it breathes out air. So
The mom's mouth? Didn't find it there. What are the rabbit pups looking for when the mom comes back? What are they getting from mom? Milk. They found it in the milk. In the milk. So in the mother's milk is this little compound, and they could take this stuff, and they could take cow milk, which does nothing to these pups. They could add that stuff, and they could get the rat pups to suckle. The rabbit pups to suckle. So that, my friends, is what a pheromone is. It's a compound that communicates a message, a simple message that elicits a simple behavior. In this case, mom is coming, milk is coming, and she had a little bit of milk on her teats, right? So that answer was exactly in the right direction. So milk carries a pheromone, and it elicits this behavior. So this is the first pheromone ever identified for mammals. We're mammals, and it's been 2 years, 3 years, almost 4 years, and we don't know much more about other pheromonal communication between mammals. Most of the work is done in insects. But I think it's an exciting thing that there could be other chemical cues that send information across different species. This was a comical, so I showed you a picture of the cockroach pheromone. It's this complicated quinoline molecule. Here's a long-chain fatty acid for the chemists in the audience. And people discovered that there's only so many chemical compounds that animals can make, right? You're an animal, you're going to make something that's going to send some signal. They figured out that this little moth and this giant elephant end up using exactly the same pheromone, which was, I think, a good calculated risk if you're communicating, because these guys don't live in the same place, and it'd be implausible that they would mate anyway, right? But so there's a little bit of, the cues can be, when this paper came out, it got so much press that these animals of huge, here's the actual size of the moth relative to the elephant, that they've happened upon the same chemical solution to signal, I am a female blank, I'm a female elephant, or I'm a female moth. In this case, crosstalk is not an issue. What else do we have? So I think one of the most interesting questions that arises in pheromone communication, we made the list, and the one thing that we didn't come upon is the kind of obvious trait that you don't want to mate with genetically, you don't want to mate with your brothers and sisters, right? Because you get inbreeding, it's very genetically deleterious. So it's important for animals to be able to discriminate self from non-self when they're choosing a mate. You could imagine animals getting mixed up and not being able to recognize siblings. You want to be able to recognize your siblings. So there's a nice story that shows that mice can actually do this. So mice, somebody mentioned, the woman in the red sweater mentioned that you want to look for, when you're choosing a mate, an animal that has the capacity to adapt, whose genes have the capacity to adapt. And in a sense, mice are doing a version of this because when they choose a mate, they're choosing, they can actually use their sense of smell to discriminate mice that are related to them separate from mice that are not related. And here's the experiment. So there's something called the major histocompatibility complex, about which I'm not an expert, but it's a recognition system in our immune system that allows us to recognize invading microbes and invading proteins they carry, and it's a unique signature, so everybody will have a slightly different MHC complement of genes. It's what makes us unique. So if you go in for, it's something that could be used as a marker to distinguish everybody in the audience has the potential to be grouped into, different MHC classes. So what people observed about 20 years ago is that mice can smell each other's MHC. So you can take a mouse and you can take a female mouse who is receptive to mating, and you can give her a choice. She walks down a tunnel and she can turn left or she can turn right. And if she turns left, she goes to someone who has the same MHC that she does. If she turns right, a different MHC. And with a very high rate of discrimination, they will choose, when they're in estrus, when they're fertile, they will choose, they don't get it 100% right, but they will choose to spend time, or they will choose to turn down the tunnel that corresponds to a different MHC type, which is pretty cool because it's a way that you can use your sense of smell to smell someone's chromosomes, which is, I think, coolest thing. That it's not, there's no visible expression of the MHC. You can't look at someone and say, I know that you're H21B. There's no way to tell by looking at someone, but mice have figured out a way to do it. And it depends where they are in their lives. So when they're in estrus, which means that they're fertile, they will choose someone that's different from their genotype. So they'll choose not to mate with their brother, as it were, but to mate with somebody very different from them. However, when they're lactating, right, when they've gone through the pregnancy, they've had their pups and they're lactating, the choice reverses. So when given a choice, they can choose different roommates in a mouse housing situation. They can choose a nestmate who's like them or a nestmate who's different from them. In that case, they choose for the same MHC type. So I want you guys to think about why are they using, they're using their sense of smell differently. When they're in estrus, they choose against MHC, and when they're lactating, they choose for the same MHC. Why is that, why are they doing that? This is just, there is no real answer, but any ideas? How about up there?
It's just a guess, but maybe once they're lactating, you've already given birth and they want to be housed with their offspring.
Right, but so the experiment is the mouse gives birth and she has her offspring, and then she has a choice of who does she want to have as a roommate. She could have somebody with the same MHC who is likely to be closely related, or someone with a different MHC. So do you want to, with that setup, why is she choosing someone with the same MHC when she's nursing her pups?
Don't her pups have the same MHC because they're related to her?
Her pups have the same MHC, but then she's given a choice of two roommates. Roommate A has the same MHC, and roommate B has a different MHC. So why is she choosing roommate A?
Because it's probably, I don't know.
Anybody else?
The composition of the milk is more similar, I would suppose.
I think I'm not setting up the experiment properly for you. So here she has a choice of two mating partners. She's choosing between two males, genetically related male, genetically dissimilar male. She chooses genetically dissimilar male. Here, let's say she's choosing boy roommate. She has a male mouse who's similar to her, or a male mouse that's different. Why is she choosing a male mouse that's similar to her when she's raising her family?
Well, in the first experiment, you want to mate with someone that has different genes to ensure like a greater gene pool,
right?
So that there aren't as many genetic defects. And in the second situation, you want people with the same, I guess, genetic composition will probably be more supportive. I guess a more supportive environment.
This is everybody's interpretation of this experiment, that when you go out into the world at some danger to yourself, away from your family, to mate, to get genetic diversity, but then you come back home when you're nursing. So the roommate who is closer to you genetically is more likely to be a close family member, less likely to eat you or your children. So that's always been my interpretation of that result. That you'll modulate your behavior. When you're mating, you're looking for genetic diversity. When you're at home, you're looking for relatives who are not going to eat you. And mice are constantly eating their pups, right? It's constant. There's so much cannibalism in the mouse world. It's horrible. But humans do the same thing. This is the awesome thing. So people said, mice can smell each other's MHC. Let's see if humans can do it. Are humans smelling each other when they choose a mate? Here the experiment is different. You rate on a 10-point scale. How pleasant is the odor of a T-shirt worn by male A or male B? And you look at two different types of women who are actively cycling, which means that in principle they're fertile. And you compare that to the choices of women who are not cycling, they're taking contraceptives, which means that they're in a state of pseudo-pregnancy, right? So let's simplify it. Here are fertile women. Here are women who are pseudo-pregnant. And in this case, the women are choosing a different genetic, they find men, the odor of a male that's different from them, more attractive. And then it switches. When you ask women who are taking contraceptives, they now prefer the same MHC type. So the point is that the graphs look the same, right? The mice go out in the wild and look for a genetically different mating partner, and then they come back home, and humans do the same thing. So there's a big increase in preference in women who are taking contraceptives. These experiments have some obvious problems. It's like two different groups of women. There's all sorts of, you should be skeptical of these kinds of experiments with humans because, yes?
So this works for humans, right?
Yes.
Because you said that, I guess societally, we don't really pay attention to pheromones so much anymore. But if it works like this and it's kind of instinctually built in, couldn't you say that pheromones still kind of work? Or, I mean, obviously they do work, but why don't they work on, I don't know, the same level as most mammals?
Yeah, I think it's a big question in the field. So no one has, 2003 was the first pheromone identified, that rabbit mammary, the milk pheromone. It's been 3 years. The minute that scientists come to the table with the compound that humans secrete that acts like a pheromone, the field will have some credibility. At the moment, these are really imperfect experiments, right? You're asking women to rate how pleasant is the smell of a T-shirt, which is not a very scientific. It's the best that we can do. The minute that someone purifies a pheromone compound from either a male or a female human and then shows that the pheromone will do something that you can measure scientifically, the whole field will have much more credibility. I would say that we behave largely as if as pheromones don't modulate our lifestyle. We don't choose partners we don't think that we choose based on these kind of animal decisions. But I would argue that there's a residue of this kind of chemical communication still in our genomes, that women are able to smell MHC, that we can select for genetic diversity when we mate. And this is like the most exciting thing to me in this field is, will we find a human pheromone? And can we go back and look at how the world has evolved and learn something about human evolution that's been driven in part by these pheromones that have these very potent effects in animals? Any other questions on this part? Professor?
They seem to play up the role of musk and things of that nature.
That's right. So we had 6 of our brave audience members smelled this, which smelled like this stuff, which smelled either like nail polish remover or caraway or what else? So this is a substance called androstenone. If you type androstenone into any search engine, you'll come up with 100,000 products you can buy that this is meant to be a pheromone that will modulate behavior. I think the jury is still out. There are some experiments, a kind of a cute experiment where you invite 50 women to participate in a study. You put a little filter paper of that stuff underneath a chair over there, and you invite the women into the room to sit, and all of the women will sit around the filter paper as if, they don't know the filter paper is there, as if it's having some sort of subliminal effects on behavior. But again, that's a terrible experiment. How do you know there wasn't something? It's hard to control. Maybe the chair looked nicer, right? It's hard to control those experiments. I think the internet is full of claims that these compounds modulate human behavior. The science is still a bit far behind, but I'm optimistic that some good scientists will step up and show that there are human pheromones, that we are in part paying attention to each other's smells. We have another question over here.
Don't look at me. Hi.
Hi.
So there's no experiment about pheromones, but I was reading, there was a study that women who go to an all-girls school or board, their menstrual cycle will be in sync.
I'm glad you asked, because this is
for
called,
those you don't know, it's a period.
So this is called menstrual synchrony, and I'm glad you brought it up. I didn't have a slide about this. So if you take any rodent, if you take a bunch of female rodents that have they menstruate at different times, you put them in a cage together, within a couple days, they are all synchronized. So there's a one dominant female who will say, somehow communicates, this is the cycle we're all going to live by. Those who are a little behind will slow down, and those others will speed up, and within a couple of days, a couple of weeks, all of the mice are cycling at the same time. And there's a residue of this in humans. So women who are co-housed, human females who are co-housed, will have the same effect. So they will begin synchronizing their estrous cycle. It's really common in animals. And that brings me to the next question of why do we do it? Why is that an adaptive behavior, and why do humans, it makes less sense for humans, but let's say why do animals do it? Why would it be useful to have 50 females all becoming fertile at the same time? Why is that useful? Any ideas? Since you brought it up, do you have any ideas? And you can forget about the human female, because it's a little icky to talk about it, but let's say, why would it be useful for 50 mice living in the same colony, for them all to be fertile at the same time? From the point of sexual selection and competition, let's say.
Well, I could think of something, but it's not why would that be good?
Well, tell me whatever you're thinking.
But if they get all pregnant at the same time, aren't they going to fight for the one mouse that's got to?
So this is good. So it's useful. So if they all get pregnant at the same time, it is useful because mice will cross foster their children. So if you have 60 pups born to 3 females on the same day, one can take some time off and they'll nurse each other's kids. So that's one very good reason why you want to have people pregnant at the same time. What's another reason? From the male point of view, why is it useful?
Well, there's more genetic diversity because the male can choose whichever female he wants, not just the ones that are fertile at that particular point.
That's right, but there's an element of competition. So why is... I think there's an element of competition in it. So you're saying the males are choosing.
Yeah.
So why is it better for the females to... If there are 2 fertile today and 5 fertile tomorrow, why is that good from the female's point of view?
Survival of the fittest. There's which one is more, I don't know. Which one is more adapted to society and which one will survive?
It's true. It puts much more competition. If you have 30 females that differ in whatever the qualities we obviously they're alive, they're female, they're the right species, but there are other qualities: stronger, faster, healthier, more attractive. You increase the focus of the competition. If everybody is fertile at the same time, that means that in a kind of a sick way, only the strongest will win the competition. If there's one male and he can only mate with so many females, the ones that have fewer, that are a little bit weaker, will lose out there. So it's a way to, it's a kind of it's a competition for males that breaks out, and that's why it persists in humans. I don't think people understand why it persists, but humans also synchronize their estrous cycles. All right. Good. So I wanted to talk about this other, I wanted you guys to imagine that you're a fish for a moment. So we imagined that we were, so what special problems do fish have, and why would fish use pheromones to solve them? This is kind of a useless picture, but okay, so anybody know about fish? How do fish choose mates? How does fish sexuality work? How about somebody over there?
Well, they drop their eggs and sperm into the water.
Right.
So I guess whenever the egg and sperm meet up, then you have a zygote and the baby will form in the water.
Excellent. So what has to be, is there an element of timing there that has to be worked out?
Well, I guess you could say that with like tide flow and I guess where they release their sperm and egg. So
right. So could
yeah.
like if the female laid her eggs today, could the male come tomorrow? No.
Not really, no.
Right. So it ends up being sort of instantaneous in most cases. Like they kind of get it together. So how could you use pheromones to cause this synchrony? Yes.
Don't most fish go back to their birth spot to lay their eggs? So maybe
there's a little.
some
Yeah, so that's
pheromones there involved.
There's a little bit of that, and that will give you some synchronization because they'll go up at the same time, so they'll all sort of meet at the same time. But even so, they'll hit dams that weren't there. There'll be some timing problem, right? Even with salmon, even if they agree to meet, plus or minus 2 days, they still have to kind of synchronize. The male and the female have to be ready at the same time. So how could you use pheromones to get it right?
Maybe the pheromone, like when they release it into the, maybe when they release the pheromone and let's say the female releases it and the male picks up on it, then maybe that's like whichever male is closest will be the one to fertilize the eggs.
True, but we're going to back it up. There's also a kind of a they choose each other first because the male's not always fertile, right? So she has to mature her eggs. So they have to go through a cycle just as mammals do, where they have to mature they're not ready to drop the eggs at any time. They have to mature their eggs, and the male has to mature the sperm, and they have to be ready at the same time, and then they have to synchronize. So how could you use pheromones to achieve that?
The female will release a pheromone to signal to the male that she's ready to mate.
Yes. So that's exactly how it works. It's a terrible figure from Tristram's book, but basically, when you take a pregnancy test, the ovulation state in human females relates. You can measure that by measuring out the hormones in the blood or the urine, right? So we secrete hormones that can give you information about are you pregnant, aren't you pregnant, are you ovulating, and fish do the same thing, but they live in water. So when they urinate, they're not urinating onto an EPT kit, they're urinating into the water, and the female will then signal the maturation state of her eggs into the water. The male listens into that, and he'll begin producing the signals that get him to mature his sperm. Then they can do their mating dance, and over about a 24-hour period, they begin doing their sexual behavior, and then they do, as our colleague up there said, they synchronize the dropping of the eggs and the sperm. And none of this could happen just by visual cues, because the male could do whatever dance he wants, but the female has to have eggs and he has to have sperm. And so this is a great way to get the biology. They're urinating anyway, but now the urine has information in it, right? The urine has cues that get the male to behave appropriately, and there's communication back and forth. So this is a great example of how chemical communication co-opted some waste signal, some hormone that was urinated out by fish, into a very effective way that you get baby fish, right? Without this, if there's even a 5-minute delay, or if the male's not ready with his sperm, then it's not going to happen. And there's some theories that this that it happened long ago, that this was built up over the time, that the female was always urinating out female hormones, and the male was oblivious to them. Then something happened where the male actually could detect that. He was actually found, he got some odorant receptor or some taste receptor that somehow allowed him to sense it. And then only those males that had that detector and were able to figure out what it meant were the ones who were able to evolve to be ready and get the response going. So that's one theory about how you would convert some non-informative urine cue into something that is selected for in evolution. Does that make sense, you guys? It's just a theory. It's hard to go back. Any questions? How were you able to get fish in this phase when they weren't queued up? Anybody want to answer that? Let's say in early times the male and the female couldn't figure out if they were fertile. Yet there are fish today. I don't know the answer, but let's see what you have. How about in the middle there?
When eggs mature, there's some hormonal changes in the fish, and these hormones, pregnancy tests right now for humans, do you test the urine and you see the presence of hormones and you see whether or not you're pregnant? I think it works the same way. Your urine contains some hormone products, but at that time, the males don't detect it, and then later on you've evolved the mechanisms.
Yep, I got it. But the question is, for the mechanism to evolve, there had to be fish that were around early on. So how did the fish at this early time create children if they didn't have this neat communication? That was the question that was posed. I'm getting you guys to answer it. But before they could smell each other's fertility, how were their baby fish created?
Is there any way it could have started out as a mutation in which one fish could do it? So then, since he could detect the pheromone,
Totally, yeah. That's totally what this slide is about. It's like this guy got a spontaneous mutation that allowed him to smell it. He's like the one fish that could smell it, and his are the progeny. But the question that remains is, how did they do it before they had a system?
It could have happened by chance.
Could have happened by chance, and that's the answer I favor. There were a few fish who were kind of hanging around in an area where there were not that many currents, and it was very inefficient, but a few females dropped eggs, and occasionally there was a male around, and it kind of worked, but it worked really inefficiently. And it was random. You couldn't really select for anything. It just was, you had few fish. That, I think, would be my answer, that it worked okay. It had to have worked because there are fish, right? That's the problem about evolutionary arguments. They're unsatisfying. All right. So I'm going to tell you a few other little neat things that animals, this is like an Animal Planet show now, another neat things that animals can do to advertise if they're good. We know that we can all look at each other and make judgments about, is he or she strong or tall, or all these physical traits that we can look at to say, this is attractive, this is not attractive. And animals do the same thing, and they do it in they're choosy. So this is a cool story. So this is a plant, which is eaten by this moth, and it has this nasty compound in it, which is we were talking about bitter taste hours and hours ago, so this is a very bitter compound. It's a toxin. Present on plants, and these male moths eat it, and they convert it into this compound. And the more of this stuff they eat, the more of this structure on their tail gets decorated with little crystals of the stuff. Eating the plant changes their actual physiology and their appearance. So the more of this plant they eat, the more of these bizarre little fronds that they're showing at the back of the male moth. And the benefit to the female is that when this male inseminates the female, he'll donate a little bit of this bitter alkaloid when he inseminates her, and then her eggs will be coated with this stuff. And it's benign to the eggs, doesn't hurt her eggs, but it will scare away any predators. So by choosing a male that's eaten a lot of this plant, she will get a sort of a gift, a courtship gift when he inseminates her. The more of the stuff he eats, the bigger this bizarre posterior display is, the more her eggs will be protected from the parasitic animals that come in that try to eat her eggs. And so we look for things when we look at potential partners. Females looking at how big is this display at the back of this male's abdomen, because it will tell me how much of this chemical I'm going to get when I mate with him, and it will protect my children. So that's it's a weird story of how plants are communicating with insects, and insects are waging a war against other insects. And they're doing it by communicating it in this very direct physical way. We know that there are alarm pheromones that are, whenever you're unfortunate enough to be bitten by like a wasp or a social insect and you smush it, they will release a pheromone that will call in all of the other wasps or bees. So why would that be a useful signal to be able to send? So we talked about sex pheromones. Now I'm going to talk about other kinds of pheromones. So we talked about this, which is a sort of a courtship pheromone to advertise how much stuff the male has eaten that he can give to her. Here's an alarm pheromone. So why are alarm pheromones, why is that a useful thing to communicate if you're a wasp or a bee?
Safety.
So explain for safety. Can you speak into the microphone?
If that's a younger alarm bee or whatever, the wasp, then the parents will come in and protect the younger one.
So it's a good way, because they're social insects, it's a good way to communicate across a pretty long distance that someone's in trouble. Why not use, we went over other possible ways, why not just use a sound to communicate that, like help, help. Like why use that?
It's discreet, so then the other, it's discreet and only that type of species would know about it.
Exactly, so it's really discreet, so only other bees and wasps can sense it. And then there's another reason, like often you'll kill this individual, but the alarm pheromone still comes out, that wouldn't work if the bee or wasp were forced to call for help. If it's injured, it can't call for help, but the pheromone does the job. Good. And then here's another really bizarre, we talked about urine has signals. Here's a really goofy, animals are so weird. So urine is full of weird cues. So this is a weird little South American rodent. The male mates with multiple females, and he'll mark the females with his urine. And we talked about urine being full of cues that are selective. So he can go around and basically put a chemical tag on different females. We've all seen dogs mark territories. This is a bizarre example of the same kind of marking where you mark a potential female. Love this picture. Everybody freaks out and laughs at this picture. It's hilarious. So why is this a useful technique?
It could show the other males that the female has already been claimed.
Right. Why is it better than, you could think of other ways of doing it. Why is this, why choose, you might imagine you could just, other animals just make the females live around the males is another, they'll defend the territory. Why is this a good way to communicate that she's mine?
Well, I doubt the rodent could protect its own territory, but it's a very strong smell, so other males can basically smell it from far away, saying that, this is a certain area that's dominated by a particular male.
Mm-hmm. I agree with that. Here's another potential idea.
I'd say each rabbit might have its own specific type of scent, so there'd be a lot more specificness.
Right, so the specificity is there, so I think all these ideas are right, that to physically fight other males for the females takes a lot of energy. If there's an agreement that you mark your physical territory, you mark your spousal territory, it requires less physical energy to defend it. There's some other hands up there.
You could go pissing on a lot of rodents, like a bunch of them.
Can you, is the mic, sorry?
Huh? You could go pissing on a lot of other rodents too. Like you could piss on that one and claim that one, and then piss on the next one, and then the next one.
I gotcha. Yeah, it's a way to. I think we're gonna have to move on in a second, but The idea is right. And the key word here is polygynous, right? So these are males that claim multiple females. It's much less energy to mark them with urine than to have to defend the territory by fighting them. Now, for something a little bit more G-rated, we have the desert ants. So you live in northern Tunisia. You're a desert ant. You come out it's a little bit like the rabbit problem. You live in northern Tunisia. It's very hot, and your job is to go out, find a source of food, and then make it back to the nest without being burned up by the sun. So what kinds of cues would you use?
A trail pheromone?
A trail pheromone, exactly. So trail pheromone. So why not use? Other ants will use landmarks. So why not use landmarks if you're a desert ant?
Because it would lead the other ants to the food. It wouldn't just say that that's where it is, but it could lead. Yeah.
But so the problem is that you live in Tunisia. You live in the desert. You have your underground nest here, and then you have to search for a couple hundred meters for a dead insect that's been burned up by the sun, and you need to bring it and grab it back and bring it back to the nest. So how do you find your way back to the nest? And why not just use physical landmarks, like look up at the rock there or the...
If you have a trail pheromone, you don't have to search for it. You just know where it is.
Exactly. So I think that those are the benefits of the trail pheromone. But other ants will use physical landmarks. Why not use physical landmarks if you're a desert ant? Anybody else up there, maybe?
You give away the location of the nest to everyone else in the desert?
True. Yeah. I mean, if you had a big showy entrance, then that would get other animals to come eat the nest if there were anteaters in the desert. But why for a trivial reason wouldn't you use physical landmarks in the desert?
It takes energy to go back to the nest and actually tell your other ants that it's over there and these are the landmarks that you need to use to get there. If you were to use a pheromone, then since it's hot, it would disperse quickly and it would reach the nest quicker than you actually traveling back.
Right. Mm-hmm. What are the physical landmarks in the Sahara Desert? I guess is my next question. I'll take that as a group answer. So you can't use physical landmarks because there may be like a little kind of a shifted area of sand, but you come back in 5 minutes and it's going to have blown away. So this is an area where trail pheromones are the only way to go. Plus a little bit of navigation. They do a little bit of navigation according to the light of the sun. And then the leafcutter ants do a lot of this. So in Brazil, leafcutter ants make enormous trails that are marked by trail pheromone. For all these reasons that only the leafcutter ant can follow it, it lasts a long time, it's cheap to produce, and it isn't, you get no eavesdropping from other animals. So I'm going to, in the little last part of the talk about how do we receive pheromones? And if humans receive pheromones, how do we do it? Which organ do we use to smell pheromones if we smell them? So we know sort of a lot about how animals do it, although it's getting kind of controversial. So here's a diagram of the cross-section of a cat. We talked about the tongue, obviously where taste happens. We talked about the nasal epithelium where smell happens. Then there's another specialized organ called the vomeronasal organ, which I'll just abbreviate VNO, because I don't like saying vomeronasal often, rapidly. So the VNO is this little specialized organ that most vertebrates have. Humans do not. This has been the sad thing, that we don't have a VNO anymore. So we have a little vestigial VNO, but it has no neurons, and it's not connected to the brain. So that's this big healthy organ that rats and cats and garter snakes have, humans have lost. And it's this organ that seems to be really important in sensing pheromones. So if you take a mouse that has no sexual experience and you remove its VNO, it will never figure out how to mate. You put it in a cage with an estrus female who's receptive, nothing. You leave him in there forever. He will never figure out how to mate with her, even though she is displaying all the signs. However, if you allow him to mate once, so he has the experience of doing so, and then remove the VNO, he's fine. So it seems that cues need to be detected early in life experience, which are absolutely essential to get the behavior. You get animals that are functionally sterile because they can't receive the cues, which I think is a cool thing. So it's this organ which humans don't have anymore, which animals have, which is absolutely required for the first time that an animal mates. And it has a very privileged way it connects to the brain. It connects to the brain, to the sort of subconscious center, so animals aren't really aware that they're sensing it, but it will elicit these interesting behaviors. And insects have a variation of this. So I showed you that movie of the silk moth, male going toward the female. So if you look deep into the brain, here's the sort of a diagram of an insect's head. This is, in the middle here is the brain. And here's the part of the brain that receives smells. If you look at the differences between a female and a male, there are some obvious differences. So anybody want to point it out to me? You can use the laser pointer. So what's different? Mm-hmm. Excellent. So it's pretty obvious that these little bits here are basically the same between males and females, but males have all this stuff that females don't have at all. There's all these brain regions that the male moth has, this giant thing, that giant thing, that giant thing, these little things, completely absent in the female. So if you were the person, I showed you the movie that the male moths respond to the female pheromone, you're the person who dissected these brains, what's your hypothesis about what that part of the brain does? Anybody? Guy in the purple?
Detects pheromones.
That's an excellent hypothesis, and how would you test it?
You let the antenna receive pheromones and see what it does for both the male and female.
Excellent, right. So you could make a preparation here. You could blow on pheromone, and you could see if those little bits light up, for instance, and that's exactly what was done. You guys are great. That's great. So there's a real dimorphism. Males smell pheromone, females don't, and you can see it in the brain, which makes sense. The brain makes sense, right? It does its job, and it makes sense. And recent advances in Linda Buck's lab, the woman who won the Nobel Prize, and Catherine Duloc's lab at Harvard. I hear several of you are going to Harvard. You can go work in Catherine's lab, have figured out that there are, again, receptors that detect pheromones. This has been the big excitement. We have all this phenomenology. You can put a mouse, you can put 50 mice, female mice in a cage, and they'll synchronize, and it's a nice observation, but where do you take it, right? You can do all these observations, but where do you take it scientifically? How do you prove that there are pheromones? You need to do chemistry to identify the pheromones. You need to do molecular biology and genetics to really figure out what are the pathways and the genes and the neural networks in the brain that allow us to smell pheromones. So that's where the field is now. And so again, this simple experiment, I told you this is the VNO. So people look for, let's find a gene that's expressed only in the VNO. It should be a pheromone receptor, right? It's a simple hypothesis. They found them and they called them V2R, the second class of vomeronasal receptors, V1R, vomeronasal receptors. It turns out that these receptors bind MHC. They bind MHC peptides. Is that not awesome? If we go back to this idea that mice can smell each other's MHC content, there are receptor proteins that sit in the VNO that can smell MHC molecules. So it really is a direct smelling of people's individual identities. So this cartoon mouse can use these receptor proteins to bind to MHC molecules that actually smell the genome of a potential partner. I think that is so awesome. And we don't know what these proteins bind. These are also membrane proteins. They may be binding to the kind of the rabbit mammary pheromone, or other small molecules that are still to be identified. And there's a little bit of work going on in the fruit fly, which is the only place in insects where you can do genetics. And if you zoom in on the head of a fruit fly, there are neurons here in the antenna that respond to all of these different proteins, bind different smells, different fruit odors. These are the receptors I told you about that bind CO2. And all of these proteins are thought to bind different pheromones. So this is the exciting thing. Can we figure out how to study the fruit fly and use it to understand how fruit flies use pheromones to go through that mating dance I talked about at the beginning? And I think I'm done, because I think that we're done. So I'm going to tell you, just at the very end, that I started my lab in December 2000. There were 4 of us. And our mission was to study the sense of smell in flies, mosquitoes, and humans. We've done pretty well. The lab grew, so by 2004, and Yelena, I guess, was a student of my friend back there. So she was a James Madison High School student who was a member of our high school outreach program and just graduated with a Ph.D. last year. And then the lab got even bigger this year, and many of these people contributed to stories that I wasn't able to tell you about today. And I've had a large group of people, I've had a lot of high school students come through my lab, and I encourage any of you, I know a lot of people want to go to medical school. Medical school is great. I would encourage as many of you who have an interest in science to check out a laboratory, because the only way that you can figure out whether science is really interesting is to do it. Nobody learns art by studying art history. You learn art by doing art. You learn science by doing science. So do science in your high schools, or find a lab in the New York City area and work in it. Science is expensive. These are the people who paid for our research. And we need to give some applause to some people here, because Jim Carrazza doing the video, Jim Carrazza. And then Todd Wells, the tongue guy. It was his tongue in the video, Todd Wells. Dejun Wan helped me with the design, Gloria Phipps. But before we go, there should be a Q&A. You guys ask me some more questions, right? Because I'm supposed to talk for a little bit longer. Any more questions?
Do you think our loss of VNO is the reason why we prefer synthetic scents over natural pheromones, or did it happen the other way, that we started using the synthetic, then we adapted to losing our VNO?
I think that this is a really controversial area of smell, but there's evidence that, because I told you that mice who have mated once can mate again. So the idea is that pheromones can also be sensed by the regular olfactory epithelium, and there's evidence that humans may have pheromone function in the regular olfactory epithelium. So I think it's possible that humans lost their VNO function because we didn't need it as much as animals, because we use visual cues to find mates, and that through lack of use we lost it. And I don't know how it connects to our love of natural scents or artificial scents.
I think I can. Thank you. Hello?
Hey, the guys who are staying, can you be quiet?
Okay. Going back to sex pheromones.
Yes.
I was just wondering, for organisms or species that are homosexual, do they prefer different pheromones or produce different pheromones from their own gender or species?
Are you talking, I guess you're talking about parthenogenic species that don't sexually reproduce. I don't know, I don't think that anything is known about that at all. I don't know anything about.
So there's no known defect in their pheromone production or preference, I guess?
Well, I mean, this is a controversial, you've hit upon a very controversial idea, right, in human biology. So about between 2 and 10% of humans, depending on how you ask, are or have been homosexual, right? And so we don't really have our finger on the biology of that. And it hasn't been, there is one study that says that the substance I had some of the audience members smell is rated differently by homosexual men than heterosexual men. And that if you do brain scans, that they will respond to it in ways that are more similar to the way a heterosexual woman will respond. This was a very famous study showing that the brains of homosexual men and heterosexual women respond very similarly, which makes sense because they both have male partners, right? So that's the only science we have. There's not much science there. What science supports a biological difference there?
Okay, thank you.
What if you have like hermaphrodites, like that can either mate with themselves or have males mate with them? Like, would they still have pheromones, or would that be just like by chance?
So C. elegans, which is a big laboratory organism, they are self-fertilizing hermaphrodites, and they have pheromones, but not all pheromones are sex pheromones, right? Your pheromones can be trail pheromones or alarm pheromones, but self-fertilizing hermaphrodites have, they still have other pheromones. They'll have aggregation pheromones, which keep members of a species together, but they don't need to use pheromones because they self-fertilize, so it's not an issue.
Some animals can like change gender, and does that have anything to do with pheromones, or is that just like adaptive, or like to have more mates, or I don't know.
This is an interesting topic for global warming because there are some animals that will change their gender depending on the temperature conditions, and it can be an all or none switch, and it can be very bad for a species, right? If the temperature raises and there are no males anymore, right, it's a very bad thing. There are other cases of reef fish who start out as female and then end up being male.
Yeah.
I don't think anyone has looked at the pheromones involved in the switch, but they behave like females first and then males, so I would presume that the whole network of things would switch.
Okay.
Do pheromones only work on like in the levels that we discussed today, or do they also work on like the cellular level?
Pheromones definitely work at the cellular level, so when they enter the vomeronasal organ, they will affect the neurons that are prepared to receive them, so the neurons will become depolarized, and then they will send messages to the brain, and those messages will, in the mouse, change the hormone balance. So you can get estrus or fertility synchronized by the pheromones. So they do much more than they can affect the biology of the whole animal.
But what about let's say like I heard from a teacher of mine that you have like cells in the heart and that they communicate, and then like if you have them in a petri dish, they'll regulate on their own. Do they secrete pheromones also?
I mean, sure. If you took pheromones, would only they work on the organ that receives the signal, right? But once you separate the organ from the body, the signal can't be sent, if that makes sense. So the way that pheromones affect the whole body is through a network of the physiology. Once you remove it, the only thing you get in a dish would be the pheromone activating the VNO, but it wouldn't cause hormone changes, right?
Humans have 5 basic tastes, you told us, but like, what about other animals? Like, dolphins, do they have 5 tastes too, or do they like missing some or have different tastes?
So the number of tastes, so it's been looked at, so insects have bitter taste, they have salt taste, they have sour taste, they have sweet taste. No one has yet shown that insects have MSG or umami taste. I don't know that anyone has looked. The problem with science is that not everything is studied in all species, so I know that fish have a different kind of a taste. They'll taste bile acids, prostaglandins, amino acids, but they don't really respond as much as I know to conventional taste.
So they have different systems than...
Well, they sense different types of compounds because sugar and bitter is less relevant to fish. So I'm not an expert on fish taste, but I don't believe that they have those conventional senses. Let's do 3 more questions and then we can all go home. So let's see, how about over there?
Yeah, you said that human beings have a vestigial vomeronasal cavity?
Yes.
How are human females able to detect the presence of MHC if this doesn't function and the receptor is
Very
in there?
good question. So as I was trying to respond earlier, there's evidence of a lot of crosstalk between the regular nose and the VNO. So even mice can detect MHC molecules with their regular nose. So it seems like over the course of evolution, There's been a backup system. The regular nose has the capacity to sense MHC. That's an excellent question. And it's believed that humans, if they can do it, they're doing it through the nose does everything, and the VNO is vestigial. How about back there?
You were talking before about the fish giving off the pheromone with the male and the female. What if there were more males in the area and they all responded to the pheromone and started producing sperm, and there was only one female?
It's a problem, and so the way the fish deal with that problem is most male fish are extremely physically territorial, so they don't use chemical communication with the other fish. They will fight with them. So what they will do first is basically establish a territory and get all the other males out, because it's a huge problem. Because there's the other problem: if you have other males, then the idea is to get the eggs fertilized with your sperm and not the other guys. So that they take care of that with fighting, not pheromones. How about one more?
Back with the ant before in the desert, you
Yes.
said that he left the pheromones for the trail, and you said he didn't use structures because the sand might cover over as the sand moves, right? So won't the sand cover over his pheromone trail?
Yeah, so the desert ants use a combination of the short-ranging pheromones, and then they also use navigation cues, so they'll navigate according to the polarized light from the sky. So you're absolutely right. Using only chemical cues in an environment like that it's only a very short-term strategy. So those guys use both the trail pheromone, and then they navigate according to the polarized light in the sky.
So why not just use one? Why not just use the sun?
I'm going to let you. So what's the degree of accuracy that navigating according to celestial cues, and what's the problem with using only celestial cues, or sun cues, sky cues to navigate?
Over the time they move?
Well, you can compensate for that, but are there cases where you wouldn't be able to see the polarized light in the sky?
It gets cloudy.
If it's cloudy, then you're in trouble. So they use a backup system, and the accuracy is they can get it they can find the nest within 3 meters, like 10 feet, but then the chemical cue gets them to the nest. I thank you guys. It's been great. Thank you very much.